For most of human history, the world felt vast, mysterious, and alive in ways our ancestors could sense but never fully understand. They watched storms roll over the horizon, felt the warmth of decaying soil beneath their feet, and witnessed life sprouting from what appeared to be nothing. Yet they never saw the quiet architects working beneath every leaf, every breath, every drop of water. This opening journey begins in that invisible realm, the world of microbes, a world so ancient, so foundational, that everything humans have ever been came long after their reign had already begun. 4 billion years ago, before continents formed or skies turned blue, before animals crawled or trees reached upward, microbes shaped the earth with patient, imperceptible precision. They built the first ecosystems, sculpted the atmosphere, and created the conditions life needed to flourish. Theirs was not a life of dominance, but one of collaboration, bacteria exchanging genes like messages, archaea stabilizing extreme environments, fungi weaving silent networks beneath soil. In this unseen cradle, the blueprint for all complex life was drafted. Even today, each human carries echoes of that ancient world, not only in our DNA, but in the trillions of microbial partners living inside and around us. And yet, we rarely think of them. Modern life renders the microscopic easy to dismiss. We wash, sanitize, polish, sterilize, actions born of necessity, but also of forgetfulness. Humanity largely believes itself self-sufficient, standing apart from the natural systems that shaped our biology over tens of thousands of years. But beneath that illusion, microbes still write the rhythms of life. They digest our meals, train our immune system, produce essential vitamins, and influence everything from inflammation to emotion. We are not solitary organisms. We are composite beings, ecosystems wrapped in skin. Imagine a Paleolithic dawn. A small band of early humans moves quietly through tall grass, dew clinging to their ankles. They are unaware that with every breath, millions of microscopic allies enter their bodies to guide development and defend them from harm. When they drink from streams, gather berries, share roasted meat, or sleep on the forest floor, their microbial partners shift and adapt in response. Early humans do not consciously choose their microbial companions, the world chooses for them. Every landscape carries its own invisible community, and every community shapes the humans who walk through it. This silent exchange is not metaphorical, it is literal symbiosis. The earliest humans did not merely coexist with microbes, they were shaped by them. Their immune systems evolved through constant microbial negotiation. Their digestion adapted to microbial chemistry. Even their moods and social behaviors were subtly influenced by microbial activity in the gut. Microbes were not passive passengers but active participants in the human story, guiding our ancestors with biochemical whispers we are only now learning to interpret. As we travel across this prologue, the focus widens. We observe ancient oceans brimming with microbial mats that produce the oxygen we breathe. We watch soil bacteria collaborate with early plants to transform barren rock into fertile land. We follow microbial spores riding on the wind, carried across continents, long before any human footstep marked the earth. Life's foundation was never built by giants but by these microscopic artisans, each one performing a function as essential as any organ in the human body. When Homo sapiens finally arrived, they were not stepping into an empty world waiting to be shaped. They entered a planet already curated by microorganisms. Forests, rivers, animal populations, nutrient cycles, all were microbial constructions. Humans simply inherited the landscapes microbes carved. And with that inheritance came dependence. Our survival hinged on their presence, their balance, their generosity. Every successful hunt, every nutritious meal, every childhood that grew into adulthood depended on the health of microbial partnerships humans could neither see nor fully comprehend. Yet despite this reliance, the relationship remained fragile. Microbes adapt quickly, sometimes in beneficial ways, sometimes in harmful ones. The immune system evolved as a constant dialogue, a conversation between host and microbe, tolerance and defense. But for most of human history, the scales remained steady. Both sides benefited. Microbes gained habitat and nourishment, humans gained resilience, diversity, and stability. This prologue quietly foreshadows what is to come, the gradual unraveling of a relationship 100,000 years in the making. It sets the emotional foundation for understanding how deeply intertwined humans and microbes once were, and how profoundly that connection has been altered by modern life. It reminds us that the story of humanity cannot be told without the story of the invisible multitudes who walked beside us, lived within us, and shaped our evolution in ways we are only beginning to grasp. As the final moments of this opening section fade, the viewer is left with a sense of reverence, a humbling recognition that the human body is not a standalone masterpiece, but a cathedral built by millions of unseen craftsmen. And before the narrative moves deeper into the timeline, one quiet truth settles over the scene: to understand how the ancient symbiosis ended, we must first understand how miraculous it was that it existed at all. Before the first stories were carved into cave walls, before language took shape in the minds of early Homo sapiens, an older and deeper story was already unfolding, a story too small for human eyes to see, yet vast enough to shape our evolution. This was the story of our first alliance with microbes. Not an agreement spoken or written, but one formed through necessity, survival, and time itself. When early humans walked through ancient forests and savannas, they carried inside them a growing constellation of microscopic partners, each one performing roles essential to their survival. And without fully realizing it, humanity entered a pact that would last 100,000 years. Picture a small group of early humans clustered around the remains of a hunt. They tear strips of raw meat, chew fibrous roots, and swallow tough, wild greens. Their bodies alone cannot unlock the full nutrition from these foods, but microbes can. In the depth of their intestines, microscopic chemists break down complex plant fibers into energy-rich molecules that the human body could never produce alone. In return, these microbes gain shelter and a constant supply of nutrients. Neither species consciously chooses the relationship, yet both benefit immensely. This mutual reliance becomes the foundation of the ancient pact. As humans face shifting climates, migrating herds, and unpredictable food sources, their microbial partners evolve with them. A change in diet reshapes the microbial communities. A shift in region introduces new microbes from the soil, water, and air. Humans are not solitary travelers, they are ecosystems on the move. Every step taken across prehistoric landscapes carries millions of microbial passengers, all adjusting, exchanging genes, adapting to new environments alongside their human hosts. But the pact extends far beyond digestion. Microbes also begin training early human immune systems. In the harsh landscapes of the Paleolithic world, danger comes not only from predators or starvation, but from invisible threats, parasites, harmful bacteria, viruses. To survive, early humans need a balanced immune response, strong enough to fight infection, but not so aggressive that it harms their own cells. Microbes become the mediators of this balance. They signal to immune cells when to stand down, when to prepare for defense, and how to respond with precision rather than chaos. Through constant exposure to diverse microbial life in soil, water, animal remains, and even on their own skin, early humans develop a resilient immune system capable of handling a world filled with pathogens. This training shapes not just survival, but childhood itself. Infants of early humans encounter microbes from birth, through soil, through food chewed by their mothers, through contact with other members of the group. These early exposures help program their developing immune systems, teaching tolerance, strengthening resilience, and preventing overreactions that could be deadly. Even the simple act of being carried against a parent's skin transfers countless beneficial microbes, creating a microbial legacy passed down generation to generation. It is an inheritance more ancient than language, more intimate than culture. And slowly, something remarkable begins to happen. Microbes do not merely live inside humans, they begin influencing human behavior. Early humans who carry microbial communities that promote calmness, better sleep, or improved stress response may survive longer and reproduce more successfully. Over countless millennia, this subtle influence shapes emotional stability, social cooperation, and even risk-taking behaviors. Microbes that help their hosts form stronger social bonds or better navigate stress indirectly ensure their own survival. In this way, microbes become quiet architects of early human psychology. The pact also extends outward into the environment. When humans forage, hunt, or settle temporarily in an area, their microbial signatures imprint onto the landscape, a faint microbial footprint woven into soil, tools, shelters, and foods. In exchange, the landscape leaves its microbial fingerprint on the humans who pass through it. Every stream they drink from carries new microscopic life into their bodies. Every animal they butcher exposes them to unfamiliar microbes. This continual exchange creates a dynamic, interwoven relationship where humans are constantly reshaped by the microbial world and vice versa. But unlike later eras, early humans do not try to control or sterilize their surroundings. There is no concept of clean versus dirty in the modern sense. The natural world is their home, and its microbial diversity becomes essential to their biology. Exposure to soil microbes strengthens immunity. Fermenting foods accidentally introduces beneficial bacteria. Variability in diet encourages rich microbial diversity. Life is dangerous, yes, infections are common, but overall microbial exposure forms a protective shield rather than a threat. This ancient harmony builds a body designed for a world full of microbes. Early humans carry a microbiome capable of breaking down wild plants, detoxifying natural chemicals, resisting infections, and regulating inflammation. Their microbial communities are diverse, robust, and perfectly matched to the unpredictable challenges of prehistoric life. Each individual's microbiome becomes a living record of the places they've traveled, the foods they've eaten, the people they've touched. It is an invisible story written inside them, changing with every new experience. And so, without ceremony, the first pact solidifies. Microbes depend on humans for habitat, stability, and nutrient flow. Humans depend on microbes for survival, adaptability, and resilience. This mutualism is not optional, it becomes central to human evolution. Over thousands of generations, microbes help sculpt the human digestive system, immune system, and even aspects of the brain and nervous system. The alliance shapes our species in ways far more profound than early humans could ever imagine. There is a quiet beauty in this interdependence. Humanity grows, thrives, spreads across continents, but always with microbial partners guiding their internal world. The pact is stable, balanced, and self-sustaining. It is one of the longest and most successful biological partnerships on Earth. But the seeds of change, of future rupture, are already planted. As humans evolve larger social groups, experiment with new tools, and explore diverse ecosystems, the delicate balance of their microbial world responds in kind. For now, the alliance holds strong, but the story of how this pact will eventually break is already waiting on the horizon. When early humans learned to command fire, they believed they had simply gained warmth, protection, and a way to transform raw meat into something safer and easier to chew. But the flames did far more than cook food, they forged an entirely new microbial universe inside the human body. Fire was not just a technological breakthrough, it was a biological revolution. And it marked one of the first major moments when humans and their microbial partners began reshaping each other in ways neither fully understood. Imagine the first sparks catching dry grass, the crackling of a small flame in the hands of hominins who had never before controlled such power. As meat roasted over the fire, its proteins loosened, its fibers softened, and its pathogens weakened. Cooking made food more calorie-dense and easier to digest, meaning humans could extract far more energy than ever before. But with this shift came a transformation in the microbial landscape of the gut. Raw meat required robust microbial communities capable of dealing with parasites, tough connective tissue, and the complex chemistry of uncooked flesh. Cooked meat, on the other hand, placed far less burden on these microscopic partners. And over time, genes, microbes, and digestive organs evolved in response. The transition to cooked food allowed early humans to reallocate internal resources. Less energy needed to be spent digesting raw plants and raw meat. This change supported the growth of larger brains, organs that consume extraordinary amounts of fuel. Microbes adapted as well. Species specializing in breaking down raw materials gradually became less dominant, while others that thrived on softer, more processed foods expanded. The gut became a dynamic, shifting ecosystem reflecting the changing nature of the human diet. The microbial world inside humans was no longer shaped solely by the environment, it was now shaped by culture, by technology, by fire. Cooking also introduced something new: consistency. Raw diets varied dramatically from day to day. A handful of berries, a scavenged carcass, bitter roots, the microbial communities had to constantly shift to handle whatever food entered the body. But cooked food was more predictable, more uniform. This stability allowed certain microbial lineages to establish stronger, more reliable populations, ultimately influencing metabolic pathways in profound ways. The ancient pact between humans and microbes deepened, becoming more intertwined as each meal reshaped the microscopic environment within. Yet not all consequences were beneficial. Fire changed exposure pathways as well. With cooking, some microbes that once entered the human body through raw foods disappeared from the diet altogether. Others, found in smoke, soil around hearths, and charred remains, became new additions. Humans spending long hours around fire inhaled airborne microbes, fungi, and particulate matter that altered respiratory and skin microbiomes. The shift from a life lived fully outdoors to one anchored around communal fire circles changed the microbial interactions of daily life. Gathering around fire also shifted social patterns. Humans now shared cooked meals in tight, intimate groups, increasing microbial exchange between individuals. Hands touching the same roasted bones, children sitting close to elders, shared tools, shared shelters, all of this created dense microbial networks among members of a tribe. The microbiome became not only individualized but communal, a shared signature of group identity. Microbes that thrived in heat-resistant environments or on cooked foods became hallmarks of early human societies, while those dependent on raw diets diminished over time. This culinary transformation also influenced the emotional and cognitive relationship humans had with food. Cooking brought pleasure, aroma, and ritual. And microbes, always silent, always present, adapted alongside these new cultural developments. Some microbial species began assisting in breaking down cooked starches, releasing energy faster than ever before. Others evolved to help detoxify compounds created during cooking. The human gut became a melting pot, not just of nutrients and enzymes, but of microbial innovation. The introduction of fire also reduced the load of harmful pathogens in food, which dramatically altered selective pressures on the immune system. Without constant battles against parasites and dangerous bacteria from raw meat, the immune system adjusted. Microbes involved in immune modulation had to evolve as well. New partnerships formed between immune cells and microbial species that thrived in the cleaner, more controlled dietary world fire created. These alliances helped prevent inflammation, repair tissue, and maintain balance within the gut, a balance humans rarely notice until it goes wrong. But this new power brought new trade-offs. While cooking helped shape a more energy-efficient digestive system, it also signaled the beginning of a slow decline in microbial diversity. Species that had supported digestion of tough, fibrous raw plants became less essential. Others disappeared entirely as the gut environment shifted. Humans gained the ability to eat food more efficiently, but lost parts of the rich microbial diversity that once characterized their early ancestors. Still, fire was a gift far greater than its losses. It allowed humans to thrive in colder regions, protect themselves from predators, and preserve meat for longer periods. It transformed social life, storytelling, and shared identity, all of which indirectly affected microbial exchange. Every new environment humans entered brought new microbes into the pact, and every cooked meal subtly redefined the terms of that invisible agreement. Over generations, microbial communities adjusted to this new lifestyle, forming a gut ecosystem unique to fire-using hominins. This set the stage for profound evolutionary shifts, from brain expansion to changes in teeth, jaw strength, and digestive efficiency. Even behavior and culture were influenced as firelight extended waking hours and reshaped social structures. And microbes were part of this evolution every step of the way. In this section, the viewer comes to understand that fire was not just a cultural milestone but a microbial one, a turning point where humans and microbes renegotiated their relationship. Cooking allowed humanity to break free from some constraints of nature, but it also restructured the internal biological world forever. This was not the end of the pact, but a major rewriting of its terms, leading to new strengths, new vulnerabilities, and new possibilities. When early humans first stepped beyond the familiar boundaries of their African homelands, they carried more than weapons, tools, and stories. They carried entire microscopic worlds within them, millions of microbial companions shaped by the soils, waters, plants, and animals of ancient Africa. As they crossed deserts, climbed mountains, and braved frozen plains, these internal ecosystems transformed with every new landscape. Migration was not a solitary journey of humans, it was a migration of microbiomes, an unfolding partnership between species both seen and unseen. The initial push out of Africa was slow, cautious, and deeply intertwined with environmental pressures. Drought cycles forced humans to search for new water sources, leaving behind the microbial richness of familiar rivers and forests. As they moved into new territories, the microbes they encountered changed dramatically. Every handful of soil had its own microbial identity. Every animal carcass exposed humans to unfamiliar bacteria. Every new plant introduced novel microscopic chemicals. With each step, the microbiome adapted, discarding some species, gaining others, reshuffling its balance to handle foreign foods and new climatic conditions. For those moving into the Middle East, the warm, arid climate selected for microbes capable of thriving in dry environments. Hunter-gatherers who settled in cooler Eurasian forests needed microbial allies skilled in breaking down seasonal berries, roots, and game meat. In the tundra, where diets became dominated by animal fat, microbes specialized in lipid digestion rose in prominence. Humans and microbes co-evolved with breathtaking speed, each migration route generating a slightly different internal microbiological identity. This diversity built resilience. Groups living along coastlines developed microbiomes shaped by shellfish, seaweed, and saltwater bacteria, organisms that helped them extract nutrients from marine life. Those in high-altitude regions fostered microbes resilient to low-oxygen environments. Meanwhile, populations that moved into dense forests encountered fungi-rich ecosystems, absorbing spores and microbial species that reshaped their immune responses. No two groups carried the same internal worlds, because no two landscapes offered the same invisible companions. The movement across continents also reshaped external microbial exposure. As humans followed migrating herds or explored new valleys, they interacted with unfamiliar animals and their microbiomes. Predators, prey, scavengers, and insects all contributed to microbial exchange. The skin, lungs, and digestive tracts of humans continuously encountered microbial signatures unique to each region. Sometimes these new microbes offered protection, aiding digestion or strengthening immunity. Other times, they brought deadly pathogens that tested the limits of human resilience. But microbial migration wasn't only a matter of survival, it influenced culture itself. As small groups settled in different regions, they developed unique food preparation methods, foraging strategies, and communal practices. These traditions became powerful forces shaping microbial diversity. A tribe that fermented roots or dried meat in a particular way unknowingly cultivated specific beneficial microbes. A group that relied on certain plants introduced different digestive challenges. Even the ways they stored food, gathered around fires, or shared meals affected which microbial species thrived in their guts. Across continents, human bodies gradually became archives of their migrations. The microbiome recorded the landscapes they had touched, the bacterial imprints of marshlands, the fungal spores of forests, the digestive enzymes from marine diets. Each individual carried a living history of their journey, encoded not in symbols or language, but in microscopic communities inside their intestines. This made early humans not just travelers, but vessels of evolving microbial knowledge. Migration also altered how humans interacted with each other. Groups that met after long separations shared microbes through touch, trade, interbreeding, and cooperation. Every handshake, every shared tool, every communal meal exchanged microscopic life. These interactions blended microbial traditions, creating hybrid microbiomes that reflected both ancestry and environment. Gradually, humanity became a network of microbial lineages weaving across continents, mixing and diverging with each new movement. Early humans likely never realized that their bodies were laboratories reacting to every change in climate, geography, and diet. They saw only the visible challenges: harsh winters, unfamiliar predators, scarcity of food. Yet their survival depended just as heavily on microscopic negotiations being carried out within. As humans entered colder climates, microbes that specialized in fermenting undigested proteins became more essential. As they encountered new pathogens, immune-modulating microbes adapted to help balance defense and tolerance. These internal shifts allowed humans to endure environments that would otherwise have been impossible. This journey created enormous diversity among human populations. Far from a single, human microbiome, our ancestors carried thousands of regional microbial ecosystems across the globe. The microbial communities in Arctic hunters differed drastically from those in tropical rainforest dwellers. Each biome shaped a different digestive capability, a different immune landscape, even subtle variations in metabolic rate and stress resilience. Microbes became local experts, teaching human bodies how to survive the specific challenges of each region. Yet despite the isolation of continents and the vast distances between human groups, one truth remained constant: the pact between humans and microbes held strong. Even as species composition shifted, the relationship remained symbiotic. Humans provided habitat and nutrients, microbes provided survival tools. This partnership allowed humans to conquer new environments without needing massive genetic changes. Microbial adaptation filled the gaps, acting as a biological bridge in places where human evolution alone would have been too slow. As this section draws to a close, the viewer begins to sense the incredible complexity of humanity's global expansion. Migration wasn't simply a movement of bodies, it was a movement of ecosystems. Every human carried within them a microbial map of the world, a living record of the climates, dangers, and foods their ancestors encountered. These invisible passengers shaped not only human physiology but also cultural identity, social dynamics, and evolutionary success. It becomes clear that long before modern borders, long before nations or civilizations, humans were already linked through a shared microbial fate. And as they continued their journey across continents, the ancient pact evolved with them, adapting silently, patiently, preparing for a future that neither humans nor microbes could foresee. In the long stretch of prehistory before agriculture reshaped human existence, hunter-gatherer life produced one of the richest and most diverse human microbiomes ever to exist. Each day in this ancient world was a dialogue between humans and their environment, spoken not in words but in microscopic exchanges. The hunter-gatherer microbiome was not an accident of biology, it was a reflection of lifestyle, movement, diet, climate, and constant contact with the living world. Every bite of wild food, every handful of soil, every animal skinned or shared around a fire left microbial traces that formed a unique internal signature of ancient life. Imagine a morning in the Paleolithic. The air is cool, carrying the scent of damp earth and crushed leaves. A small group wakes in a temporary shelter woven from branches. Before the sun fully rises, they are already gathering roots, tubers, nuts, and berries, foods raw, fibrous, and complex, requiring microbial assistance to unlock their nutrients. These plants are covered in natural bacteria, yeasts, and microbes from the soil. Without refrigeration, pesticides, or industrial cleaning, humans consume these organisms with every meal. Far from harmful, many of these environmental microbes strengthen immunity, promote digestion, and increase microbial diversity within the gut. Hunter-gatherer diets are unpredictable, shifting not only by season but by day. One day may involve hunting large game, rich in fat and protein. The next might bring nuts, fruits, or bitter greens. Another might offer nothing at all. Microbes respond to this unpredictability with remarkable flexibility. They expand and contract in response to nutritional availability, reshaping metabolic pathways to extract as much energy as possible from whatever food enters the body. This metabolic versatility becomes one of the defining traits of the hunter-gatherer microbiome, an internal ecosystem built for resilience, capable of switching between famine and feast without collapsing. But diet is only one element of this ancient microbial world. Daily contact with soil, water, and animals also plays a central role. Children crawl on the ground, their hands coated in dirt rich with bacteria that help train their immune systems. Adults dig for tubers, forage through underbrush, and process raw meat from freshly hunted animals. This constant exposure to diverse microorganisms acts as a form of natural vaccination. It prevents immune overreaction, strengthens tolerance, and promotes a balanced inflammatory response, a benefit that modern humans, shielded by sanitized environments, often lack. Movement also shapes the microbial signature of hunter-gatherer life. These groups rarely stay in one location for long. They migrate according to seasons, food availability, and climate changes. Each new campsite introduces a new set of environmental microbes. The gut ecosystem shifts slightly with every location, gradually accumulating a portfolio of microbial experience. Over time, this creates immense microbial richness, far greater than that of sedentary modern populations. The more landscapes humans touch, the more microbial partners they gain. Social behavior further amplifies this diversity. Hunter-gatherers share everything: meals, tools, shelters, and physical space. A hunted animal is divided among the group, and everyone eats from the same fire, passing bones, skins, and tools from hand to hand. This communal lifestyle encourages constant microbial exchange. A microbe that thrives in one person's gut may transfer to another, creating a shared microbiome reflective of group identity. This shared microbial signature becomes as integral to community life as shared rituals or ancestry. Fermentation appears early in human history, often by accident. Leftover berries ferment naturally. Meat stored underground develops microbial layers that preserve it. Tubers sometimes soak in water for days, allowing beneficial bacteria to break down toxins and make them safer to eat. Although primitive, these accidental forms of fermentation introduce probiotic microbes into the diet, organisms that boost digestive health and support immunity. Over generations, hunter-gatherer groups unknowingly cultivate microbial allies through these simple food preservation methods. Hunter-gatherers' physical activity also influences their microbial world. Hours of walking, climbing, hunting, and gathering promote metabolic pathways that interact with gut microbes in unique ways. Exercise increases blood flow to the gut, changes hormone levels, and creates conditions that certain microbes thrive in, particularly those associated with endurance, stress regulation, and inflammation control. The microbial environment of a constantly moving hunter-gatherer differs significantly from that of a modern sedentary lifestyle, which often reduces diversity and encourages inflammatory imbalances. Seasonality adds another layer of complexity. In summer, diets may revolve around fruits and fresh plants, feeding fiber-loving microbes. In winter, diets shift toward preserved meats or high-fat animal foods, nurturing entirely different microbial groups. These seasonal oscillations keep the microbiome dynamic and adaptable. Far from being harmful, this continual fluctuation becomes a biological advantage, a training system that prepares the gut for constant change. Even childbirth influences microbial inheritance. Babies born in primitive environments acquire microbes from the mother's birth canal, skin, and breast milk. They crawl in soil, taste stones, and touch everything within reach. These behaviors, seen as dangerous in modern contexts, were essential training rituals for the immune system. Childhood in hunter-gatherer societies was a microbial apprenticeship. The combined effect of diet, movement, environment, and social living creates a microbiome far more diverse, resilient, and balanced than anything seen in modern populations. This ancient microbial signature supports strong immunity, flexible metabolism, and emotional stability. It is a living snapshot of what humans evolved to carry inside them, a complex ecosystem reflecting 99% of human history. As this section closes, the viewer begins to appreciate how deeply the natural world shaped early humans from within. The hunter-gatherer microbiome was not just a biological feature, it was a profound expression of lifestyle and environment. It was an invisible companion that adapted, learned, and evolved alongside us. And although this ancient microbial richness has largely disappeared in modern societies, its memory remains encoded in the evolutionary foundations of our biology, reminding us of a time when humans lived in seamless harmony with the microbial world. The dawn of agriculture stands as one of humanity's greatest turning points, a transformation so sweeping that it redefined not only how humans lived, but how their bodies functioned on the inside. For hundreds of thousands of years, humans had moved with the seasons, foraging and hunting their way across ever-changing landscapes. Their microbiomes reflected this diversity, shifting with each new environment, each new food source, each new climate. But when early humans settled into permanent villages and began cultivating crops, the ancient contract with microbes quietly began its first major revision. What had once been a fluid, dynamic microbial world started narrowing, simplifying, and reshaping itself around an entirely new lifestyle. Agriculture began gradually, not as a sudden revolution but as a series of small experiments. Seeds saved from wild grasses were planted near campsites. Animals lingered around human settlements, scavenging waste until eventually becoming domesticated companions. Slowly, humans learned which crops thrived in certain soils, which irrigation methods yielded the most food, which plants could withstand drought or pests. As they became skilled cultivators, their diets changed dramatically. Where once they consumed dozens of plant species and wild foods each season, they now relied heavily on a few staple grains: wheat, barley, millet, rice, maize. This narrowing of diet had profound microbial consequences. Grains, especially when consumed in large quantities, fostered different microbial communities than the varied fibrous plants of hunter-gatherer life. Microbes specialized in breaking down complex fibers began to decline, while those adapted to starch-heavy diets increased. Over time, the gut microbiome became less diverse, more uniform, and more dependent on domesticated food sources. This loss of diversity wasn't immediately noticeable, but it marked the beginning of a subtle biological shift, one that would echo through millennia. Sedentary living further complicated the microbial landscape. Permanent villages reduced the constant environmental exposure that once shaped the hunter-gatherer microbiome. Instead of roaming across forests, plains, and rivers, humans now interacted mostly with the same patch of land day after day. The soil around settlements changed as well, shaped by human waste, domesticated animal droppings, and the repetitive planting of crops. This altered soil microbiome inevitably influenced the microbes humans encountered through food, water, and daily contact. Domesticated animals became new microbial partners, and new sources of danger. Living in close quarters with goats, sheep, pigs, and chickens meant constant exposure to animal microbes, some beneficial, some harmful. Zoonotic diseases, pathogens capable of jumping from animals to humans, became more common. Before agriculture, humans rarely lived close enough to animals for such exchanges to occur frequently. Now, barnyards became hotspots of microbial exchange. Diseases like smallpox, measles, and tuberculosis trace their origins to this era, emerging from microbes once exclusive to animals. Storage of food also introduced new microbial dynamics. Grain harvested in large quantities had to be kept for months, sometimes years. Moisture, pests, and fungal spores infiltrated these storage areas. Humans consumed food laced with molds, yeasts, and bacteria they had never encountered in such concentration before. Some of these newcomers aided digestion or enhanced immune function, while others brought toxins and illnesses. The gut microbiome had to adapt once again, adjusting to the microbial risks of a sedentary farming life. Agriculture also brought cooking methods that further changed microbial exposure. Porridges, breads, and boiled grains replaced raw plants and fermented foods. These cooked meals, more processed and softer, required less microbial assistance for digestion. Slowly, microbial species that once thrived on raw fibrous foods dwindled. Meanwhile, microbes that specialized in breaking down simpler carbohydrates began to dominate. This dietary transition shifted metabolic pathways inside the human body, altering blood sugar regulation, energy storage, and hunger rhythms. Another major shift came with population density. Hunter-gatherer groups were small, often fewer than 50 individuals. Farming villages, however, grew into hundreds or thousands of people. With so many individuals living close together, microbes spread more rapidly. Colds, intestinal infections, and skin diseases circulated through crowded settlements. The immune system had to adapt to this new microbial environment, often becoming more reactive and more inflamed as a response to constant exposure to pathogens. Water sources changed too. Instead of drinking from flowing streams, people relied on wells or standing water. Runoff from animal pens, waste pits, and farming fields contaminated these water sources, introducing new bacteria and parasites into the human gut. Farmers developed microbial partnerships that helped manage these pathogens, but the constant immune activation took a toll. And yet, agriculture also introduced new beneficial microbial traditions. Fermentation techniques emerged independently in multiple regions as ancient peoples discovered that buried, soaked, or aged grains and vegetables transformed into foods that lasted longer and tasted better. These early fermented foods, sour porridges, primitive beers, yogurt-like dairy, became powerful sources of probiotic microbes. In this way, humans unknowingly worked alongside microbes to preserve their food and enhance its nutritional value. This cultural innovation temporarily restored some of the microbial diversity lost through agricultural diets. But despite these benefits, the overall trend was clear: the microbiome of early farmers was narrower, less adaptable, and more vulnerable than that of their hunter-gatherer counterparts. The body that had evolved for a life of constant movement, environmental exposure, and diverse diets now faced monotony and crowding. The ancient microbial pact entered a new phase, not broken, but undeniably altered. Humans still relied on microbes, but the range of microbial allies available to them had diminished. This narrowing of microbial diversity carried significant evolutionary consequences. Immune systems became more reactive, smoothing the path for the rise of allergies and inflammatory diseases in future generations. Metabolic pathways shifted under the influence of starch-heavy diets, laying the groundwork for conditions like insulin resistance. These changes would not fully manifest until modern times, but agriculture planted the seeds of future biological challenges. And yet, for all its complications, agriculture allowed humanity to flourish in numbers never before seen. With stable food supplies, larger communities, and more complex social structures, humans spread across the world at unprecedented rates. Microbes evolved alongside them, adjusting to this new lifestyle, renegotiating their roles in the human body. The ancient symbiotic contract had survived the shift, but it was no longer the same agreement. The world of microbes within humans had begun to shrink, becoming less wild, less diverse, and increasingly shaped by human choices rather than natural environments. The next chapters of history would deepen this shift, setting the stage for even more dramatic transformations in the delicate relationship between humans and their invisible partners. When the first cities rose from the fertile river valleys of Mesopotamia, the Nile, the Indus, and ancient China, humanity believed it had entered a new age of progress, a triumph of architecture, farming, and social order. Yet beneath the stone walls, beneath the markets and temples, something unseen stirred. Cities did not merely bring people together, they brought their microbes together too. And for the first time in human history, the density of human life began reshaping microbial evolution in ways that were both transformative and devastating. In a hunter-gatherer world, people lived in small groups, isolated from one another for long periods. Disease spread slowly, often dying out before it could travel far. But cities changed the rules. Thousands of individuals lived in close quarters, sharing wells, markets, livestock pens, communal fires, and cramped sleeping spaces. Every cough, every handshake, every shared tool became a conduit for microbial exchange. Crowds meant that microbes had endless opportunities to spread, mutate, and adapt. A single infection could ignite a chain reaction, sweeping through entire populations with speed previously unimaginable. Sanitation was nearly non-existent. Waste accumulated in streets, attracting insects and rodents that carried their own microbial worlds. Human and animal refuse mixed in muddy walkways, seeping into the soil and the water. Parasites thrived in stagnant puddles. Bacteria bred in the moist shadows of alleyways. The air carried the breath of thousands, a dense microbial fog hovering over early urban life. People inhaled these invisible particles daily, and their lungs, guts, and skin adapted to the constant microbial pressure. Pathogens, once rare visitors, now became permanent residents. Diseases like dysentery, cholera, typhoid, and tuberculosis found fertile ground in the crowded, unsanitary conditions of early cities. These were microbes that had evolved rapidly to exploit the new human environment, one where large populations ensured they could spread continuously without burning out. The ancient microbial pact began to fracture under this pressure. For the first time, microbes were no longer solely allies or harmless companions, many became enemies. Yet even as harmful microbes flourished, beneficial ones declined. The diverse, fiber-rich hunter-gatherer diet had already narrowed during the agricultural era, but city life reduced it further. Urban diets relied heavily on processed grains, breads, porridges, and domesticated animal products. Fresh wild plants became rare luxuries. Microbial species dependent on rough fibers dwindled, replaced by those adapted to starch-heavy diets. The gut ecosystems of city dwellers lost resilience, becoming more uniform and less capable of handling infections. The drinking water situation intensified this shift. People depended on wells, cisterns, or river systems that were often contaminated by human and animal waste. Waterborne pathogens circulated constantly, forcing the immune system into a perpetual state of alert. Children growing up in ancient cities encountered far more microbial threats than their rural or nomadic ancestors. Some survived, developing strong immune systems shaped by repeated exposure. Others perished before reaching adulthood. Microbes became silent architects of life expectancy. Domesticated animals, now living in close proximity within city walls, played a major role in shaping urban microbes. Cows, pigs, goats, chickens, and dogs all carried microorganisms capable of jumping to humans. Some of the most devastating diseases in history originated through these interactions. The boundary between human and animal microbiomes blurred as people slept beside livestock or walked through streets filled with manure. This constant mingling introduced new microbial challenges that human bodies had never encountered before. But microbes were not merely threats, they also shaped culture and survival strategies. Early civilizations noticed that certain food preparation methods reduced illness. Cooking grains thoroughly, boiling water, fermenting milk or vegetables, drying meats, these practices emerged from necessity, guided by repeated microbial lessons. Without fully understanding the science, ancient peoples developed intuitive relationships with microbes. Fermentation became a cultural cornerstone, not only preserving food but enriching it with beneficial bacteria that strengthened gut health. Still, the biggest change brought by cities was the emergence of epidemics. Diseases that required large, dense populations to sustain themselves, measles, smallpox, influenza, could now evolve and spread. These pathogens became evolutionary specialists in exploiting crowded human environments. When immunity developed in one population, the pathogen mutated, returning in new forms. Microbial evolution accelerated, driven by the sheer number of human hosts available. The psychological landscape shifted as well. Fear of illness became intertwined with daily life, influencing rituals, hygiene behaviors, taboos, and religious practices. Entire belief systems emerged around purity, contamination, blessings, and curses, cultural responses to the invisible threats that filled city life. Microbes began shaping not just bodies, but minds and societies. Yet despite all the dangers, cities thrived. Their economic, social, and cultural advantages outweighed the microbial challenges. Humans did not retreat, they adapted. Over generations, the immune systems of urban populations changed. People who survived childhood illnesses passed on genetic advantages. Microbes that once overwhelmed entire communities gradually became manageable. Humanity and microbes continued their ancient negotiation, but now in a world of complexity and constant pressure. The microbial signature of city life became distinct, narrower in diversity, richer in pathogens, shaped by crowding, domesticated animals, and contaminated water. This signature represented a pivotal shift in the ancient pact, a transition from a world where microbes were predominantly teachers and allies to one where they also became adversaries. But even in conflict, the relationship remained deeply interwoven. Humans influenced microbes by creating cities, microbes influenced humans by shaping immunity, culture, and survival. As this section closes, it becomes clear that the rise of civilization came with a cost that would echo throughout human history. The invisible enemies that emerged in ancient cities would evolve, spread, and transform alongside humanity. And each step forward in progress would require humans to renegotiate their fragile, ancient symbiosis with the microbial world. Long before humans understood microbes, they learned to collaborate with them. Fermentation began not as a deliberate invention, but as an accident, a forgotten bowl of grains bubbling in the sun, a pot of milk souring near a warm fire, a cluster of fruit fermenting on the forest floor. Ancient people tasted these transformed foods cautiously, then curiously, and finally gratefully. Without knowing it, they had stumbled upon one of the most profound biocultural partnerships in history, a way to harness microbial power to preserve food, enhance flavor, and nourish the body in ways raw ingredients never could. Fermentation was humanity's earliest form of biotechnology, a silent collaboration between unseen organisms and human ingenuity. And for thousands of years, this art shaped not only cultures and cuisines, but the microbial landscapes within the human gut. Every fermented food, tangy, fizzy, sour, aromatic, was a vessel for beneficial bacteria and yeasts that supported digestion, strengthened immunity, and helped humans survive harsh climates and unpredictable seasons. Imagine an early agrarian village. Harvest season has arrived, and the air is filled with the scent of fresh grains and ripe fruit. But abundance brings challenges. Food spoils quickly without refrigeration. Families cannot consume everything at once. Then, almost magically, fermentation offers a solution. Grains soaked in water begin to fizz and thicken, becoming sour porridges. Vegetables submerged in brine transform into crisp, tangy pickles. Milk stirred in clay pots ripens into yogurt-like curds. These foods last longer, taste richer, and are far safer to eat. Behind these transformations lies a microscopic orchestra. Lactic acid bacteria convert sugars into acids that preserve the food and protect it from pathogens. Yeasts break down starches, creating bubbles and alcohol in early beers and wines. Wild microbial communities from soil, air, and human hands colonize these foods, creating flavors unique to each region. Different climates foster different microbial combinations. Cold mountains produce thick, tangy fermented milk, humid valleys give rise to sour pickles, coastal regions create fish sauces rich with salt-tolerant bacteria. In every culture, fermentation becomes a mirror of its environment, a microbial signature shaped by landscape, tradition, and time. As fermented foods spread across ancient communities, they began reshaping human internal biology as well. These foods introduced living microbes into the gut, boosting microbial diversity and helping early humans digest tough plant fibers and detoxify harmful compounds. In an era without medical knowledge, fermentation acted as a natural probiotic therapy, reducing intestinal infections and improving nutrient absorption. Children weaned onto fermented porridges developed stronger microbiomes. Adults who consumed fermented foods regularly enjoyed better digestive resilience. In many ways, fermentation became a silent guardian of human health. The cultural significance of fermentation grew alongside its biological impact. In China, crocks of fermenting vegetables became winter lifelines. In Africa, sorghum beer was not just a drink but a ceremonial staple, shared during rituals and celebrations. In the Middle East, fermented dairy shaped entire cuisines. In Europe, breads, cheeses, and wines became symbols of identity and prosperity. Every culture crafted its own microbial masterpiece. And through these foods, humans formed intimate bonds with the microbial world without realizing it. Fermentation also played a crucial role in moments of crisis. During long winters, fermented vegetables prevented vitamin deficiencies. In droughts, fermented grains sustained communities when fresh foods were scarce. Nomadic tribes relied on fermented milk during long journeys. Even sailors used fermented beverages to prevent illness at sea. Microbes became invisible allies in survival, stepping in where nature faltered. But beyond practicality, fermentation influenced emotion and social connection. The scent of a fermenting pot signaled comfort and nourishment. Families passed down fermentation vessels, each containing microbial lineages older than the memories of their owners. Clay pots, wooden barrels, and woven baskets held not only food, but generations of microbial culture, microscopic histories shaped by countless hands. These vessels became heirlooms, storing not just recipes but microbial ancestry. However, fermentation was not merely beneficial, it also introduced challenges. Some ferments went wrong, producing toxins or dangerous molds. Others developed unpredictable flavors. Yet humanity learned, generation by generation, how to recognize safe fermentation, how to cultivate the right microbes, how to avoid the dangerous ones. This evolving knowledge formed early food science, rooted in observation rather than laboratory theory. It taught humans a truth they understood instinctively long before discovering bacteria: transformation can be a form of preservation. As civilizations grew more advanced, fermentation expanded into new realms. Bread leavened with sourdough cultures allowed for softer, more nutritious loaves. Cheeses developed complex microbial communities that contributed unique aromas and textures. Alcoholic beverages became central to rituals, trade, and social gatherings. Fermented fish sauces, soy-based foods, vinegars, and kombucha-like beverages emerged independently across continents. It was as if humanity and microbes had co-written a global culinary language. And yet, as transformative as fermentation was for culture, its influence on the human microbiome was even more profound. The beneficial bacteria introduced through fermented foods helped counteract the declining microbial diversity caused by agriculture and early urbanization. In crowded cities where pathogens thrived, fermentation offered a counterbalance, a way to restore internal microbial harmony. Through these foods, humans replenished microbial species that farming and sanitation had slowly reduced. In many ancient regions, people consumed fermented foods daily. Their bodies depended on this regular influx of beneficial microbes to maintain balance. It was a natural form of microbiome maintenance, long before such concepts existed. But as time passed and civilizations evolved, the role of fermentation shifted. Industrialization and modern food processing would eventually push fermented traditions aside, replacing them with sterile, packaged alternatives. The microbial richness once central to human diets began to fade. What remained were only echoes of an ancient partnership, forgotten crops, abandoned cellars, and fading traditions. This section highlights not only the beauty and ingenuity of fermentation, but its profound biological importance. Fermentation was a bridge between culture and biology, between human intention and microbial creativity. It nourished bodies, preserved harvests, strengthened immunity, and shaped entire civilizations. As the narrative moves forward, the viewer begins to sense a subtle sadness, a realization that modern life has nearly severed these ancient threads. Fermentation was once a daily ritual that kept humans connected to the microbial world, but its decline marks another fracture in the thousand-century pact. The story ahead will show how this ancient art, once essential for survival, slowly vanished under the weight of progress, leaving behind a microbiome longing for its forgotten allies. For tens of thousands of years, humanity's microbial worlds changed slowly, shaped by local environments, regional diets, and ancient traditions. But when the age of exploration began, when ships set sail across unknown oceans, when continents connected for the first time since deep prehistory, the microbial world erupted into motion. Humans believed they were exploring new lands, discovering new civilizations, expanding their influence across the planet. Yet unseen, beneath every plank of wood, under every sailor's fingernails, within every barrel of food, microbes were traveling with them. The age that reshaped global history also unleashed one of the largest microbial migrations the world had ever witnessed. The first explorers stepped onto their ships carrying more than supplies. They carried microbial signatures shaped by their homelands: European gut flora built on bread, meat, and fermented dairy, Asian gut communities shaped by rice, soy, and pickled vegetables, African microbial lineages tied to diverse wild plants and fermented porridges. Inside every sailor and traveler was a biological map of where they came from. And as ships cut through waves, these microorganisms joined them on the journey, clinging to bodies, food, clothing, and cargo. Life on board vessels created new microbial environments. Ships were cramped, damp, and filled with people who rarely bathed. Barrels of water teemed with bacteria. Food stored for months developed fungal layers and bacterial colonies. Rats, insects, and livestock added their own microbial contributions. With every long voyage, microbes adapted to these harsh, enclosed ecosystems, becoming hardier, more resilient, more capable of surviving extreme conditions. Some of these ship-borne microbes would go on to influence entire continents. When explorers arrived in new lands, the microbial collisions began. The Columbian Exchange is remembered for its movement of crops, animals, and goods. But its most dramatic and immediate impact came from microbes. European diseases like smallpox, measles, and influenza entered populations that had never encountered them. Without prior exposure, indigenous immune systems were unprepared for the microbial onslaught. The result was catastrophic: entire civilizations weakened or vanished within decades. Microbes crossed oceans faster and more relentlessly than any human army. But the exchange was not one-sided. The Americas had their own microbial ecosystems: soil bacteria, plant-associated microbes, gut flora shaped by maize, beans, squash, and wild fruits. These microorganisms entered the bodies of arriving explorers, challenging their immune systems in unexpected ways. Travelers faced new infections, foodborne microbes, parasites, and fungal exposures. Some adapted, others succumbed. Microbes were not passive passengers in global conquest, they were significant players, determining who survived and how societies changed. Trade routes that emerged from exploration carved microbial highways across the globe. Silk, spices, tea, coffee, and textiles all carried invisible microbial cargo. Even objects like coins, tools, and scrolls transferred bacteria and fungi across continents. As goods exchanged hands, new microbial species entered communities, reshaping internal ecosystems in subtle but lasting ways. The global microbiome, once divided by natural geographic barriers, began to blur. Food cultures transformed as well. Ingredients introduced through exploration, tomatoes, chili peppers, potatoes, cacao, sugar, reshaped cuisines worldwide. These new diets introduced new microbial interactions within the gut. Cultures that had once depended on local, fermented foods now incorporated foreign crops, altering the microbial balance that had defined their internal environments for centuries. The gut microbiome became more globalized, reflecting humanity's expanding connections. But exploration's impact extended beyond human bodies. It changed entire ecosystems. Animals introduced into new continents carried their own microbes, influencing soil biology and plant health. Crops brought to unfamiliar regions interacted with native microbial communities, sometimes thriving, sometimes disrupting delicate ecological balances. Microbes spread into waterways, forests, and farmlands, modifying environments just as drastically as human activities did. In many ways, microbes acted as the true explorers, adapting faster than humans could travel, embedding themselves into every new place. They leaped between populations, continents, climates, and species, evolving with each new host. Some became global pathogens, others became beneficial additions to local microbiomes. Their influence grew with every voyage, shaping nutrition, immunity, disease patterns, and even cultural identity. The age of exploration also altered the psychology of microbial exposure. Sailors learned that unfamiliar foods or water sources could sicken them. Communities developed fear of foreign pathogens. Quarantine, one of humanity's early attempts to understand microbial threat, emerged from this era. Port cities adopted isolation practices long before germ theory existed. People sensed an invisible danger in the movement of disease, even if they lacked the language to describe it. Still, exploration brought microbial gifts. New fermented traditions arose where cultures collided. Europeans learned from Asian fermentation techniques. Africans introduced microbial-rich foods to the Americas. Taste, aroma, and microbial diversity spread along these routes, enriching culinary worlds across the planet. In certain places, traditional fermentation increased, helping restore some of the microbial diversity lost during earlier phases of human history. Yet the shadow of microbial exchange remained. Epidemics continued to break out in port towns, trade centers, and heavily traveled regions. Diseases shaped politics, warfare, migration patterns, and economies. Microbes quietly determined the fate of empires, just as they had shaped the rise and fall of ancient cities. By the end of the age of exploration, humanity had become more interconnected than ever, and so had its microbes. The internal worlds of human populations no longer evolved separately. They blended, merged, collided. Diversity increased in some regions, collapsed in others. The global microbiome was now in motion, responding to trade winds, ship routes, and human ambition. As this section draws to a close, the viewer recognizes that exploration did far more than map new continents. It remapped the microbial world, altering health, ecology, and civilization itself. Every major historical shift of this era was shadowed by microbes traveling unnoticed across oceans, shaping destinies from within. And in the centuries to come, human progress would accelerate even further, bringing new transformations, and new strains on the ancient pact between humans and the microscopic life they carried. The Industrial Revolution is often remembered for smokestacks, machines, railroads, and the transformation of societies from rural landscapes into bustling urban centers. But beneath the roar of engines and the glow of gas lamps lay another revolution, one unfolding at microscopic scales. The shift from agrarian life to industrial life marked the second great rupture in humanity's 100,000-year microbial partnership. Where agriculture had narrowed the diversity of human microbiomes, industrialization began to erase entire microbial lineages, altering the internal human world in ways no era before had achieved. Before industrialization, most humans lived in close contact with soil, animals, and natural environments. They consumed minimally processed foods, breathed outdoor air, touched living earth daily, and shared homes with microbes from the land. But as factories rose and cities expanded, this intimate connection with nature began to dissolve. Brick, iron, and coal replaced forests, fields, and rivers as the architecture of human life. And with every new invention that made life safer, faster, and more convenient, microbes were pushed further from the human body. One of the earliest microbial casualties came from changes in food production. Industrialized milling stripped grains of their fibrous layers, reducing the natural bacteria once present on raw foods. Sugar, flour, and processed carbohydrates became dietary staples. Canning and pasteurization, while revolutionary in preventing spoilage and disease, sterilized foods to such an extent that beneficial microbes no longer entered the gut with regular meals. Humanity had gained safety, but lost microbial richness. The rise of mass sanitation followed. Cities invested in sewage systems, chlorinated water supplies, and public health measures that dramatically reduced exposure to pathogens. Death rates dropped, life expectancy climbed, and epidemics that had terrorized cities for centuries became less frequent. But these victories came with unintended biological consequences. Clean water and sterilized environments deprived the immune system of the microbial training it once received from everyday exposure. The internal balance between helpful microbes and the immune system began to shift, leaning toward hypersensitivity and inflammation. Industrial work environments also restructured microbial exposure. Instead of farming, gathering, or hunting outdoors, people spent long hours in factories, metal-filled, chemical-laden spaces where air circulation was poor and natural microbes were scarce. Urban apartments, often stacked tightly together, replaced open landscapes. Families lived in enclosed rooms with minimal ventilation, reducing contact with environmental bacteria that once shaped robust immune systems. Children grew up playing in streets of stone and soot rather than soil and grass, weakening the diversity of microbes they encountered early in life. Another major shift came with chemical innovation. Fertilizers, pesticides, and industrial chemicals flooded the environment, altering soil microbiomes in dramatic ways. Soil once teeming with microbial life became depleted in many regions. Plants grown in these altered soils contained fewer natural microbes, meaning humans consuming them also received less microbial nourishment. The microbial chain connecting earth to food to human body weakened year by year. Antibiotics, miraculous in their ability to cure deadly infections, became both saviors and silencers. Although their widespread use is covered more deeply in later sections, industrialization laid the groundwork for their rise. As medicine advanced, humans gained unprecedented power to kill harmful microbes. But antibiotics do not distinguish between enemy and ally. Many beneficial bacterial species, shaped by millennia of coevolution with humans, vanished from gut microbiomes following repeated antibiotic exposure. Industrialized societies became dependent on these drugs, accelerating microbial loss in ways no previous era had seen. Industrial workplaces also introduced new airborne exposures. Coal dust, metal particles, and chemical fumes filled the air in factories and cities. Instead of inhaling the microbial diversity of natural landscapes, people inhaled substances that inflamed airways and disrupted microbial communities in the lungs and sinuses. Respiratory microbiomes, once shaped by forests, grasslands, and oceans, now reflected the harsh interior of industrial cities. Even childbirth practices began to change. Hospitals became the new birthplaces of society, replacing homes and natural environments. Babies born in sterile hospital rooms encountered fewer maternal microbes during delivery. Medical interventions, though often necessary, exposed infants to antibiotics, disinfectants, and sterile equipment that shaped their early microbial colonization. A newborn's first microbial partners, once drawn from the mother's skin, gut, and environment, now came from sanitized medical settings, altering the microbial inheritance passed down for thousands of generations. Urban crowding intensified microbial challenges. Millions of people lived in dense, polluted neighborhoods, often lacking fresh air or natural spaces. Industrial-era city dwellers had less contact with animals, less exposure to soil bacteria, and fewer fermented foods in their diet. Instead, they consumed factory-made meals, often lacking the live microbes once abundant in traditional diets. The gut ecosystem, already narrowed by agriculture, contracted further. Industrialization also changed emotional and psychological landscapes, indirectly affecting microbes through stress. Long working hours, unsafe labor conditions, and rigid schedules produced chronic stress responses in the body. The gut-brain axis, deeply influenced by microbial activity, reacted to this new emotional environment. Stress hormones altered the gut's microbial composition, decreasing beneficial species and promoting inflammatory responses. Microbes once supported by relaxed outdoor living struggled to survive in bodies shaped by factory schedules and urban anxiety. Despite these changes, industrialization also improved survival in ways no era before had achieved. Clean water, medical advances, and improved sanitation saved millions of lives. But these benefits came at the cost of microbial diversity that once protected humans from disease, regulated immunity, and maintained metabolic health. The ancient pact had not fully broken, but it was being rewritten at a pace far faster than evolution could follow. The most striking aspect of industrialization's microbial impact is that humanity did not notice it happening. People saw progress, sanitation, convenience, and modernity. They did not see the microbial extinctions unfolding inside them, life forms disappearing silently as new technologies reshaped food, water, birth, and environment. Microbes that had traveled with humans for thousands of years, passed down through generations, carried across continents, began to vanish without ceremony. This section closes with a realization: industrialization marked the beginning of a profound disconnection between humans and the natural microbial world. It was a triumph of engineering but a disturbance of biology. Cities grew brighter, cleaner, and safer, but human internal ecosystems grew smaller, simpler, and more fragile. The story of the next century would only intensify this rupture, as modern societies continued pushing microbes to the margins, unaware that they were dismantling a partnership essential for human health and survival. As the modern world accelerated into the late 19th and early 20th centuries, a profound psychological shift began to take hold. For the first time in human history, people no longer saw microbes as hidden partners in life's delicate balance, they saw them primarily as threats. Germ theory, though one of the greatest scientific breakthroughs ever achieved, reshaped humanity's relationship with the microbial world in ways no cultural or technological change had done before. With the discovery that unseen organisms cause disease, fear took root. And from that fear emerged a new cultural standard, the ideal of sterility. Before this shift, humans lived bathed in microbial richness. Dirt under the nails, animals in the yard, shared drinking vessels, open-air kitchens, and fermented foods were normal parts of life. Microbes were everywhere, and though some caused illness, many supported immunity, digestion, and emotional well-being. But once scientists revealed that microscopic organisms could kill, every surface, every cough, every touch became suspicious. Cleanliness turned from a luxury into a moral virtue, a symbol of intelligence, modernity, and responsibility. Soap companies, cleaning brands, and advertisers seized this moment. They painted microbes as monstrous invaders hiding on doorknobs, lurking on dishes, waiting on children's toys. Housewives were told that visible cleanliness was no longer enough. True cleanliness meant eliminating invisible germs. Hospitals, which were once chaotic and unsanitary, embraced antiseptic practices that saved countless lives. Streets were disinfected, water systems sterilized, and homes filled with chemicals designed to eradicate microscopic life. The sterile ideal spread faster than any pathogen, becoming a hallmark of modern living. But as people scrubbed away dirt and disinfected their environments, something deeper began to change. Without realizing it, humans were erasing the everyday microbial exposures their bodies had evolved alongside for 100,000 years. Infants, once surrounded by the natural microbes of family, soil, and animals, were now raised in sanitized rooms with sterilized bottles and chemically cleaned floors. Children who might once have played barefoot in mud now encountered surfaces wiped clean with antibacterial wipes. Families who once shared fermented foods and rustic meals now relied on packaged, pasteurized diets lacking living microbes. The immune system, which depends on exposure to diverse organisms in early life to develop balance and resilience, began receiving fewer signals. Without this microbial instruction, immune cells sometimes grew confused, overreactive, or poorly regulated. Allergies, autoimmune diseases, and inflammatory disorders began rising, quietly at first, then dramatically, as generations grew up with reduced microbial contact. The sterile environments meant to protect people were also depriving their bodies of essential microbial teachers. As cleanliness became synonymous with safety, cultural narratives reinforced the divide between humanity and the microbial world. Schools taught children that germs were the enemy. Hospitals displayed posters declaring war on invisible invaders. Media portrayed microbes as sources of dirtiness, danger, and disorder. The nuanced truth, that the vast majority of microbes are harmless or beneficial, faded from public consciousness. Fear overshadowed balance. Inside the home, the sterile ideal took on its strongest form. Antibacterial soaps became household staples. Bleach was poured into sinks and onto floors. Kitchen counters were wiped repeatedly throughout the day. Even the act of hanging laundry outdoors, once a natural exposure to environmental microbes, disappeared as dryers became common. With each new cleaning product, the microbial world inside the home shrank further. Beneficial species that once colonized furniture, walls, and bedding vanished, replaced by sterile surfaces that offered no microbial diversity to the body. Outside the home, urban life reinforced this separation. Concrete sidewalks replaced fields and forests. Indoor offices replaced outdoor labor. Cars replaced walking. And as people spent more time inside curated, climate-controlled environments, their exposure to natural microbes dwindled. Plants were kept in pots, soil was covered with manicured lawns, and animals were increasingly separated from daily life. The world grew cleaner, yes, but also biologically thinner. This cultural shift also influenced attitudes toward birth and infancy. Hospitals, striving for sterility, began separating newborns from the microbial exposures they would naturally receive from their mothers and homes. Caesarean sections became more common, altering early microbial colonization. Bottle feeding replaced breastfeeding in many societies, further reducing the transfer of maternal microbes that guide immune and digestive development. Babies began life with fewer microbial allies than any generation before them. Even the emotional landscape of microbial fear began shaping behavior. People began avoiding physical touch, sharing drinks, or sitting on the ground. Children were discouraged from exploring outdoors without supervision. Entire industries centered around sterilized water, sterile packaging, and antimicrobial clothing. The sterile ideal seeped into the subconscious, changing how humans saw themselves in relation to the natural world. Yet despite its shortcomings, the sterile ideal did accomplish extraordinary things. It reduced infections, lowered child mortality, and made surgical procedures safe. It improved public health on a scale unimaginable before the 20th century. But these achievements came at a biological cost humanity did not yet understand: the erosion of the ancient symbiotic relationship that microbes once nurtured inside every human body. As the world embraced cleanliness, it unknowingly stepped further away from microbial diversity, and walked closer toward the consequences of microbial loss. The immune system began misfiring, metabolism shifted, and mental health patterns subtly changed. Humans had declared war on microbes, but in doing so, they also weakened the microbial foundations needed for long-term well-being. This section closes with a quiet tension. The sterile ideal brought undeniable benefits, but it also created a fragile internal world, disconnected from the ecosystems that once shaped human resilience. The next chapter will reveal the most powerful tool humanity created in its war against microbes, and the irreversible changes it brought to the ancient pact: antibiotics, the miracle that saved millions and altered microbial history forever. When antibiotics first entered the world, they were received as nothing less than a miracle. For thousands of years, humans had lived at the mercy of microbial infections that could strike without warning, a simple cut turning deadly, childbirth becoming a gamble, and epidemics devastating entire communities. The discovery of penicillin changed everything. It was as if humanity had suddenly gained the power to command life and death at microscopic scales. But in this triumph, a new and far more complicated chapter began, a chapter where humanity's greatest medical breakthrough also became its most profound microbial disruption. The story begins with Alexander Fleming, who noticed a mold growing on a forgotten petri dish, a mold that killed bacteria surrounding it. This accidental discovery gave birth to antibiotics, substances capable of stopping bacterial infections in their tracks. The world quickly embraced them. Soldiers wounded in war now survived infections that once claimed lives. Children recovered from illnesses that had historically been fatal. Surgical procedures, once perilous due to infection risk, became safer. Antibiotics became synonymous with hope, progress, and modern medical power. But beneath this triumph lay an unintended consequence. Antibiotics are not precise weapons. They do not differentiate between harmful pathogens and beneficial microbes. Each dose delivered into the body acted like a sweeping fire, killing millions of bacterial species regardless of their role. The gut, once home to thousands of microbial lineages passed down across generations, faced sudden and repeated extinctions. Within hours of taking an antibiotic, entire microbial communities could collapse. Species responsible for digesting complex fibers, regulating inflammation, producing essential vitamins, and stabilizing mood were wiped out. Some populations recovered after treatment, but many did not. Others returned in altered states, weakened or imbalanced. Each course of antibiotics subtly reshaped the internal microbial landscape, narrowing the diversity that had been carefully sculpted over millennia. In the early decades, no one noticed these invisible losses. People saw only the benefits: the fevers that subsided, the infections that healed, the relief that spread through families as loved ones recovered. Antibiotics seemed like perfect solutions, and because they worked so effectively, they were prescribed often, sometimes too often. For viral infections, antibiotics were mistakenly used even though viruses cannot be killed by them. Livestock were given antibiotics to promote faster growth. Industrial agriculture embraced them as tools to increase food production, exposing humans indirectly to antibiotic residues in meat and dairy. All of this created unprecedented pressure on microbial life. Inside the human gut, beneficial bacteria weakened under constant assault. Microbial species that once thrived in harmony now struggled to maintain balance. Some vanished completely, leaving gaps in ecosystems that had functioned smoothly for thousands of years. These gaps were often filled by opportunistic microbes, bacteria capable of surviving antibiotic attacks, including strains that could cause inflammation, digestive issues, or long-term immune problems. Outside the human body, bacteria began to evolve defenses. Antibiotic resistance emerged, a natural but accelerated evolutionary response. Microbes exchanged survival genes, learned to neutralize antibiotics, or developed protective biofilms. The more antibiotics were used, the stronger these resistant strains became. Hospitals, once the safest places for healing, increasingly faced infections that no drug could treat. Yet the story of antibiotics is not one of villainy, but of complexity. They remain among the most important tools in modern medicine. They save millions of lives each year and enable procedures like organ transplants, joint replacements, cancer treatments, and premature infant care, feats unimaginable without them. But the cost of this power is biological. Every antibiotic taken alters the internal microbial world, disrupts ancient symbiotic relationships, and contributes to a slow erosion of microbial diversity. Infants became particularly vulnerable. Babies born in hospitals, exposed to sterile environments and sometimes given antibiotics early in life, began their microbial journey with fewer species. Their gut ecosystems lacked the ancestral richness once inherited from mothers and environments. Early life antibiotic exposure weakened microbial development during the most critical window, a time when the immune system learns tolerance, balance, and self-regulation. These changes were subtle, often invisible, yet they shaped long-term health in ways society had not anticipated. Adults, too, felt the effects. People who experienced multiple courses of antibiotics across their lives often carried less diverse microbiomes. Metabolic pathways shifted. Inflammation increased. Conditions such as allergies, autoimmune diseases, obesity, and mood disorders began to rise in industrialized nations, not solely due to antibiotics, but influenced by the cumulative disappearance of microbial allies who once helped regulate these systems. The environmental impact grew as well. Wastewater carried antibiotic residues into rivers and soils, altering microbial ecosystems far beyond human bodies. Resistant bacteria spread through agriculture, wildlife, and global travel. Microbes that had never encountered each other before shared genetic tools for survival, creating a worldwide network of resistance. Humanity's attempt to control microbes had inadvertently strengthened some of the most resilient among them. Still, the narrative is not hopeless. Antibiotics remain essential, and when used responsibly, their benefits far outweigh their risks. But this section reveals the emotional and philosophical paradox of antibiotics. The same tool that protected humanity from microbial threats also weakened the ancient microbial partnership that supported human health. Humans had become powerful enough to kill microbes, but not wise enough to foresee the consequences. The double-edged nature of antibiotics becomes clearer as the modern world unfolds. The internal ecosystems humans once carried as gifts from nature were now fragile, sensitive to every new medical intervention. Microbes that had shaped immunity, digestion, and emotional well-being were disappearing faster than evolution could replace them. The microbial world inside humans was no longer a dense forest, it was becoming a sparse garden. This section closes on a reflective note. Antibiotics solved one set of problems but quietly created another. The ancient contract between humans and microbes, once built on coexistence, was now strained by conflict. The next chapter in this story will reveal how modern diets and industrial foods further accelerated microbial loss, reshaping the inner landscape of human biology in ways no one could have predicted. The rise of processed food marked a new chapter in humanity's relationship with microbes, not through medicine or sanitation, but through the very substances people consumed every day. For most of history, food had been alive in a microbial sense. Raw plants coated in soil bacteria, fermented dishes teeming with probiotics, meats that carried natural microbial populations, and grains that still contained the yeasts and microorganisms of the fields where they grew. But with industrialization came a new idea: food that could last longer, travel farther, resist spoilage, and be manufactured on massive scales. This idea reshaped diets around the world, and with it, the internal microbial worlds that depended on natural food diversity. Processed food began as a convenience, a way to meet the demands of rapidly expanding cities and modern lifestyles. Canned goods, refined sugars, white flour, preservatives, artificial flavorings, and later fast food chains became symbols of modernity and comfort. They offered consistency, affordability, and ease. But beneath the surface, these foods carried an invisible consequence: they were nearly devoid of the microbial life that once nourished the human gut. Where a fresh apple contains thousands of natural microbes, a packaged snack contains none. Where traditionally fermented foods deliver living bacteria that support digestion and immunity, pasteurized and factory-produced foods offer sterility. Over time, these sterile diets began reshaping the human microbiome with every bite. One of the most significant shifts came from the removal of dietary fiber. Hunter-gatherers once consumed up to 100 grams of fiber per day from roots, fruits, plants, and wild foods. Even early agricultural diets contained far more fiber than modern industrial diets. Fiber is not digested by the human body directly, it is the preferred food source for beneficial gut microbes. When fiber disappears, so do the microbial species that rely on it. And with them vanish the metabolic byproducts they produce, compounds that reduce inflammation, regulate blood sugar, support immunity, and even influence mood. Processed foods, rich in refined carbohydrates and sugars, created a new environment inside the gut. Microbes evolved to thrive on these simple, rapidly absorbed nutrients became dominant. They multiplied quickly but offered little long-term benefit. The gut ecosystem, once a dense forest of microbial diversity, began to resemble a monoculture, a microbial landscape lacking the complexity needed to support robust health. Artificial ingredients added another layer of disruption. Preservatives designed to inhibit microbial growth in foods often carried similar effects within the digestive system. Emulsifiers used to improve texture interfered with the protective mucus layer lining the gut, exposing microbes to harsher conditions and altering their balance. Artificial sweeteners, though calorie-free, influenced microbial pathways in ways that affected metabolism and insulin sensitivity. Each new chemical innovation brought subtle but powerful shifts to the microbiome. Fast food accelerated this transformation. High-fat, low-fiber meals consumed rapidly and frequently conditioned the gut toward inflammation and metabolic imbalance. Beneficial bacteria that once flourished on diverse natural diets dwindled, replaced by species that thrived in the nutrient-dense, fiber-poor environment of industrial meals. And because processed food often lacked beneficial microbes entirely, the gut received no reinforcements, only challenges. The global spread of processed foods compounded these effects. Traditional diets rich in fermentation, fresh produce, and diverse plant species began disappearing as Western-style packaged foods entered markets worldwide. Communities that once maintained rich ancestral microbiomes, shaped by local ingredients and regional microbes, began losing them in a single generation. The microbial signatures that took thousands of years to evolve were overwritten by sterile, manufactured diets. Children, especially, felt the impact. Early exposure to processed foods meant their developing microbiomes did not receive the variety of fibers, natural bacteria, and fermented compounds that once guided immune training. Many nations saw rising rates of childhood allergies, digestive issues, and metabolic disorders, not as isolated problems, but as symptoms of a broader microbial collapse. Yet this shift was not only biological, it was emotional and cultural too. People began associating convenience with comfort, speed with satisfaction. Homemade meals, slow fermentation, and natural cooking methods faded as processed foods promised immediacy. The microbial rituals that once united families and communities, sharing fermented dishes, preparing meals from scratch, preserving harvests, were lost to the rhythms of modern life. Inside the body, the consequences grew clearer with each generation. Reduced microbial diversity meant less resilience. Gut microbes became less capable of protecting against pathogens, less effective at supporting immune balance, and less helpful in maintaining metabolic stability. The rise of chronic diseases, obesity, diabetes, autoimmune conditions, digestive disorders, was influenced not only by caloric excess and lifestyle, but by the gradual disappearance of microbial partners essential for biological harmony. The processed food era also altered the psychological dimension of the microbiome. The gut-brain axis, the biochemical highway between microbes and mental health, responded to these dietary shifts. Species that produced calming neurotransmitters declined. Inflammatory pathways increased. Mood disorders became more common, especially in industrialized nations with highly processed diets. The internal world shaped by millennia of natural eating habits was being replaced by one molded by factories, packaging plants, and synthetic ingredients. Despite these effects, the allure of processed food continued to grow. It was cheap, accessible, and engineered to appeal to human cravings. Salt, sugar, and fat, rare in nature, became abundant, activating reward pathways in the brain. The microbial cost remained invisible to most people. Modern society embraced processed food as progress, unaware that each meal was rewriting the ancient microbial pact in ways no generation before had experienced. This section ends with a reflective tension. Humans had gained convenience and abundance, but lost microbial diversity, metabolic balance, and biological resilience. The foods that once fed both body and microbiome had been replaced by products designed primarily for taste, shelf life, and speed. The internal ecosystems humans inherited from their ancestors were being dismantled meal by meal. In the next chapter, the focus turns to the consequences of this microbial erosion, the vanishing of ancient microbial species once central to human health, and the profound, often unnoticed loss that reshaped the very foundations of modern biology. For most of human history, the microbiome was a vast, resilient, and diverse ecosystem, a living archive of ancient partnerships formed across continents, climates, and generations. But as the modern world accelerated through industrialization, sanitation, antibiotics, and processed food, something quiet and irreversible began to unfold inside the human body, the disappearance of ancestral microbes. These were not just random bacterial species, they were evolutionary companions that had lived with humans since the dawn of our species, shaping aspects of immunity, metabolism, emotional regulation, and even behavior. Their loss did not happen in a single moment, but slowly, subtly, as modern lifestyles eroded the environmental and dietary conditions those microbes depended on. Scientists studying remote hunter-gatherer groups in the Amazon, Tanzania, and Papua New Guinea discovered a startling truth: these communities carried microbial species that had vanished from industrialized populations entirely. Their guts hosted lineages of bacteria that processed wild fibers, broke down toxic compounds in plants, stabilized inflammation, and produced metabolites no longer present in many modern bodies. These microbes were not simply scarce, they were extinct in the microbiomes of people living in cities, suburbs, and highly sanitized environments. This extinction was silent, unnoticed, and profound. The disappearance began with the narrowing of diets. Ancient microbes relied on the complex fibers found in wild plants, roots, nuts, and diverse seasonal foods. Modern diets, stripped of fiber and filled with refined carbohydrates, provided no sustenance for these species. Without food, they dwindled. Without fiber-rich environments to sustain them, they failed to reproduce. And once gone, they could not return, because the conditions needed for their survival no longer existed in the modern gut. Another major force behind their vanishing was the reduced exposure to soil, animals, and natural environments. For thousands of years, humans lived with dirt under their nails, slept on the ground, gathered plants coated in soil-based microbes, and drank from rivers and springs rich in environmental bacteria. These exposures replenished the microbiome constantly, adding new species and maintaining diversity. But with urbanization came paved streets, indoor living, filtered water, and chlorinated systems. The influx of fresh environmental microbes slowed, then stopped. Children growing up inside apartments, schools, and paved neighborhoods encountered only a fraction of the microbial diversity their ancestors once absorbed every day. Hygiene practices also altered microbial inheritance from parent to child. Babies historically received a robust microbial transfer during vaginal birth, early breastfeeding, and constant physical closeness with mothers and siblings. But rising C-section rates, formula feeding, and rigorous sterilization reduced this early microbial inheritance. Infants began life missing key species their ancestors once relied on, and without exposure to dirt, animals, or fermented foods, they had no way to acquire them later. Antibiotics accelerated the decline. A single treatment could wipe out dozens of microbial families, repeated treatments across childhood erased even more. Some microbes recovered, but others never returned. Antibiotic-driven extinctions accumulated across generations, each family losing microbial diversity that once defined their lineage. Over time, entire branches of the human microbial tree disappeared from modern populations. Processed foods further contributed to this loss. Lacking natural microbes and filled with preservatives that inhibit bacterial growth, they introduced nothing to replenish the gut ecosystem. Fermented foods, once central to human diets, became commercialized and pasteurized, reducing the diversity of live microbes they provided. Without these microbial reinforcements, gut communities weakened, leaving ancestral species with no allies and no environment in which to thrive. The consequences of this microbial extinction remain far-reaching. Many lost species were involved in regulating inflammation, a function deeply tied to modern illnesses like asthma, allergies, autoimmune conditions, and chronic inflammatory disorders. Other vanished microbes once produced neurotransmitter precursors, influencing mood stability, anxiety levels, and stress responses. Their disappearance has contributed to emotional and mental health challenges in ways scientists are only beginning to understand. Some ancestral microbes helped extract nutrients from complex plant fibers, meaning their loss contributed to rising metabolic issues despite abundant food. Others protected against pathogens, forming defensive barriers that reduced infection risks. Without these ancient protectors, modern immune systems face both understimulation and overreaction, swinging between vulnerability and chronic inflammation. Yet perhaps the most haunting aspect of microbial loss is its finality. Once an ancestral microbe disappears, it cannot simply be reintroduced. Many depended on environmental interactions that no longer exist, on wild foods, soil microbes, nomadic movement, animal contact, or fermentation traditions forgotten by industrialized societies. Their extinction mirrors ecological loss in the natural world, species wiped out not by single events, but by the gradual erosion of the habitats they needed to survive. As this section draws to a close, the viewer gains a sense of something precious slipping away, not through violence or catastrophe, but through absence. A quiet emptiness forms where microbial richness once thrived. The human body, once a thriving ecosystem shaped by millennia of coevolution, now carries only a shadow of its former diversity. The ancient microbial partners that guided human evolution, protected early childhood, and supported resilience are fading into memory. The story ahead will explore the moment when this ancient contract finally broke, not through a single invention or discovery, but through the combined weight of modern life pressing against an ecosystem never designed to withstand such change. For nearly 100,000 years, the relationship between humans and microbes had been a living treaty, an unspoken, delicate balance shaped by evolution, environment, and shared survival. But as modern life advanced, pressure mounted from every direction: antibiotics, sanitation, sterile births, processed foods, indoor lifestyles, chemical exposures, environmental loss. Each force alone could strain the microbial partnership. Together, they formed a perfect storm. And somewhere in the last century, quietly and without ceremony, the ancient contract finally broke. This break did not happen in a single moment. It unfolded gradually, a slow unraveling that few noticed. The human body, once home to thousands of microbial species working together in harmony, began losing its diversity. Vital species disappeared, communication networks within the gut collapsed, and microbial systems that had evolved alongside humanity for millennia shrank into fragile collections of survivors. Modern humans were still ecosystems, but impoverished ones, stripped of biological allies essential for regulating immunity, metabolism, and emotional balance. The first fracture came with birth. For millions of years, infants acquired their initial microbial foundation through intimate contact with their mothers, exposure to vaginal microbes during natural delivery, skin microbes during breastfeeding, and environmental microbes as they explored the world. This transfer created a living inheritance, a microbial dowry passed from one generation to the next. But with rising medical interventions, C-sections became common. While life-saving in many cases, they disrupted this natural transmission, giving newborns a radically different set of initial microbes, often dominated by hospital bacteria instead of ancestral maternal species. Formula feeding compounded this shift. Breast milk carries complex sugars and living bacteria designed specifically to feed and nurture an infant's developing microbiome. Formula, despite being nutritionally complete in other ways, lacks this microbial complexity. Infants fed primarily on formula gained fewer beneficial species and missed critical early life microbial cues that shape immune development. A body missing these early signals learns to react differently, sometimes overreacting, sometimes failing to respond appropriately. The second fracture came with antibiotics, powerful, life-saving, and biologically disruptive. Infections that once threatened survival were now easily cured, but at the cost of microbial extinctions inside the gut. Children receiving multiple antibiotics in early life lost entire microbial lineages that never returned. Adults treated repeatedly for minor infections experienced cumulative losses. Each course altered the internal world further, clearing space for opportunistic microbes less beneficial than those they replaced. Over time, these repeated disruptions weakened the microbiome's stability, making it more vulnerable to imbalance and disease. Chemical exposures delivered another blow. Industrial cleaning agents, disinfectants, pesticides, and food preservatives all played roles in reducing microbial diversity. These chemicals, designed to kill or repel microbes in environments, often had similar effects inside the body. Gut microbes sensitive to chemical residues diminished. Environmental microbes that once replenished the microbiome were wiped out before they could ever reach human hands or mouths. Even the dust in modern homes carried far fewer microbial species than the soils and open-air environments of ancient life. Processed foods further reshaped the microbial landscape. Diets rich in fiber, the primary fuel for beneficial gut bacteria, were replaced by meals high in refined sugars, fats, and additives. Without fiber, many microbial species starved. Without fermentation, humans lost natural sources of probiotic microbes. Industrial diets fed only a narrow set of gut bacteria, leading to monocultures where diversity once thrived. The gut, once a vibrant forest of microbial life, became more like a field of crops repeatedly planted and harvested, stripped of richness and complexity. Urban lifestyles added pressure. Humans moved into environments that disconnected them from nature, sealed buildings, chlorinated water, paved streets, artificial lighting. Children no longer played in mud, rarely touched animals, and spent far more time indoors. Adults worked in climate-controlled offices, breathed filtered air, and handled sanitized surfaces. Environmental microbes that once entered the body daily became rare visitors. The microbial inputs that once shaped resilience dwindled. These changes affected not only the gut but the entire immune system. The immune system evolved to coexist with microbial diversity. It required constant negotiation with allies and threats alike. But when microbial diversity vanished, the immune system found itself without proper guidance. It misinterpreted harmless substances as threats, triggering allergies. It attacked its own tissues, contributing to autoimmune diseases. It struggled to distinguish self from enemy. The ancient training system, microbes teaching immune cells how to behave, had broken down. Even the gut-brain axis began to reflect this rupture. Microbes once responsible for producing neurotransmitters, regulating stress responses, and supporting emotional stability diminished. Mood disorders, anxiety, and mental health challenges rose sharply in industrialized societies. These changes were influenced not just by lifestyle or culture, but by the quiet dissolution of microbial partners that had once helped modulate emotional balance. The final fracture came when all these pressures converged. The human microbiome, weakened by multiple generations of microbial loss, reached a tipping point. Modern humans carried fewer species, fewer genetic tools, fewer protective mechanisms than any humans before them. Microbial functions that once supported health were missing or incomplete. The ancient contract, built on coexistence, mutual benefit, and biological cooperation, could no longer hold. Humans had become the dominant force in the partnership, reshaping microbial life with medicine, sanitation, diet, and technology. But the microbiome, essential to human function, could not adapt quickly enough. Evolution works slowly, yet human society changed rapidly. This mismatch created vulnerabilities modern medicine still struggles to address. Some scientists describe this rupture as a form of internal extinction. Just as animals disappear from ecosystems when their habitats are destroyed, microbes vanished from the human body when the conditions they needed were eliminated. The body remained, but its invisible inhabitants were gone or diminished. Entire microbial clans, once guardians of health, lived only in isolated populations or had disappeared entirely from industrialized groups. And yet, despite this loss, the rupture is not absolute. Many microbial species remain, struggling to adapt to the modern world. Others survive in remote communities, preserved by lifestyles untouched by industrialization. Some linger in ancient fermented foods, waiting to be reintroduced. The pact may be broken, but echoes of it still exist, remnants of a once thriving partnership that shaped human evolution. This section ends with a sobering reflection. The ancient contract between humanity and microbes did not fail because of malice or neglect. It broke because human progress outpaced biological evolution. In building safer, cleaner, more advanced societies, humans unknowingly dismantled the microbial foundations of their own bodies. The story ahead will explore what this rupture means for modern health and how the consequences of microbial loss began shaping the age of chronic illness that defines the contemporary world. The rise of chronic illness in the modern world did not arrive with a sudden catastrophe or dramatic viral outbreak. It emerged slowly, quietly, like a shadow spreading across industrialized societies. Autoimmune diseases, allergies, asthma, obesity, diabetes, inflammatory disorders, mood imbalances, conditions once rare in human history became common in just a few generations. Doctors searched for genetic explanations, environmental triggers, dietary culprits. But beneath all these factors lay a deeper, often unrecognized thread, the disappearance of ancestral microbes that once shaped human health. The ancient contract had broken, and chronic illness stepped into the empty space left behind. For most of human evolution, the immune system worked in close collaboration with microbes. Beneficial bacteria trained immune cells to distinguish friend from foe, encouraged tolerance toward harmless substances, and prevented overactive inflammatory responses. Microbes were instructors, teaching newborn immune systems how to react, when to fight, and when to stand down. But as microbial diversity declined due to antibiotics, sanitation, urbanization, C-sections, formula feeding, and processed foods, the immune system lost its teachers. It began to misfire. One of the earliest signs of this misfire was the rise of allergies. For tens of thousands of years, humans lived surrounded by pollen, dust, animal dander, soil microbes, and plant compounds. Their immune systems learned early to tolerate these exposures. But modern children, raised in sanitized homes with limited outdoor play and reduced microbial contact, encountered far fewer environmental microbes. Their immune systems, lacking proper early life training, began treating harmless substances as threats. Allergies exploded in frequency: hay fever, eczema, peanut allergies, asthma, conditions almost unheard of in ancestral populations. Autoimmune diseases followed a similar pattern. Without microbial signals to guide them, immune cells sometimes turned inward, attacking the body's own tissues. Rheumatoid arthritis, Crohn's disease, multiple sclerosis, type 1 diabetes, illnesses shaped by internal inflammatory confusion became increasingly common. These diseases did not spread like infections, they emerged from deep biological imbalance, a system missing the microbial cues it evolved to depend on. Metabolic disorders also rose sharply. Gut microbes once played essential roles in regulating appetite, extracting nutrients from food, controlling blood sugar, and managing fat storage. They produced short-chain fatty acids that reduced inflammation, improved insulin sensitivity, and supported healthy metabolism. But processed diets, low in fiber and high in refined ingredients, caused these microbial partners to diminish. Without them, metabolism began to malfunction. Obesity rates soared, not simply because of increased calories, but because the microbial systems regulating energy balance had been weakened or lost. Diabetes type 2, once rare, became a global epidemic. Chronic inflammation, driven partly by microbial imbalance, altered how the body processed glucose. The microbiome's ability to communicate with fat cells, liver cells, and hormones was disrupted. The body stored fat differently, responded to food differently, and experienced hunger differently. These changes were not just cultural or behavioral but biological, rooted in microbial decline. Mental health, too, bore the imprint of microbial loss. The gut-brain axis, a communication network involving nerves, immune cells, and microbial metabolites, once relied on microbial diversity to maintain emotional stability. Ancestral microbes produced compounds that influenced serotonin, dopamine, and other neurotransmitters. They helped regulate stress responses and reinforced neural pathways associated with calmness and resilience. As key microbial species disappeared, this communication weakened. Anxiety, depression, and stress-related disorders grew more prevalent. While these conditions are multifaceted, part of their rise is linked to internal shifts shaped by microbiome loss. Bodies missing crucial microbial signals respond differently to stress, regulate mood less effectively, and show altered patterns of neural activity. The emotional resilience of ancient humans, shaped by constant environmental exposure and stable microbial ecosystems, no longer exists in the same biological form. Even digestive disorders reflect this internal imbalance. Conditions like irritable bowel syndrome, IBS, inflammatory bowel disease, IBD, chronic bloating, and food sensitivities became common as microbial diversity decreased. The gut, once buffered by a complex microbial community, became more reactive and less stable. Foods tolerated by humans for thousands of years now triggered discomfort or inflammation. The digestive system, deprived of essential microbial allies, struggled to perform tasks it once executed seamlessly. The environmental consequences of microbial loss compounded these issues. Industrialized societies spend most of their lives indoors, in cars, offices, apartments, and climate-controlled buildings. These environments contain fewer microbial species than natural landscapes. Humans now inhale and touch far less microbial diversity than their ancestors who lived outdoors, farmed, hunted, gathered, and played in soil. This lack of exposure contributes to immune dysregulation, making modern bodies more prone to inflammation and sensitivity. Chronic illness, in this sense, is not simply a medical problem, it is an ecological one. Human bodies are ecosystems, and ecosystems collapse when diversity disappears. A forest with many species can survive storms and pests. A forest reduced to a handful of species collapses under pressure. The same principle now applies to the human microbiome. With ancestral microbial partners gone, bodies no longer respond to stress, food, or pathogens the way they once did. Yet the most striking aspect of chronic illness is how quietly it spread. Unlike plagues or infections, chronic conditions unfold slowly, building over decades, appearing subtly at first. A child develops allergies. A teenager struggles with anxiety. An adult gains weight or experiences digestive issues. A senior develops an autoimmune condition. These patterns seem personal and isolated, yet they form part of a broader biological narrative, one shaped by the loss of microbial diversity. This section closes by reframing chronic illness not as a failure of individual bodies, but as a sign of a broken biological relationship. The ancient contract with microbes, which once ensured balance and resilience, has eroded. What remains is a human body functioning without the microbial guidance it evolved to rely on. The next chapter will explore one of the most surprising aspects of this story, how deeply microbes influenced not only the body, but the human mind itself, shaping mood, cognition, and emotional well-being in ways that modern science is only beginning to understand. Long before humans understood neurons, synapses, or brain chemistry, microbes were already influencing the human mind. The relationship between the gut and the brain, known today as the gut-brain axis, is not a modern discovery, but an ancient biological conversation, whispered across millions of years of evolution. Microbes help shape human emotions, stress responses, cognitive patterns, and social behavior long before the first words were spoken or the first tools carved. And yet, only recently has science begun to uncover how profoundly these invisible partners contributed to the architecture of human thought. To understand this connection, imagine the earliest humans wandering through prehistoric landscapes. Their survival depended not only on physical strength but on intuition, emotional balance, cooperation, risk-taking, and the ability to interpret danger. These traits were not governed by the brain alone. Microbes in the gut produced neurotransmitter-like chemicals, serotonin, dopamine precursors, GABA, that influenced mood stability, alertness, decision-making, and even courage. In this way, microbes subtly shaped behavior in ways beneficial to both themselves and their human hosts. A more socially cooperative human created safer group dynamics, beneficial for microbial spread. A calmer, more resilient human was more likely to survive stress and starvation, keeping microbes alive as well. Microbes and brains evolved together. The gut microbiome also influenced how early humans perceived the world. Certain microbial species produced metabolites that affected pain sensitivity, helping humans push through physical hardship. Others influenced sleep cycles, allowing better rest during long migrations. Some microbes even affected memory formation, helping humans remember where edible plants grew or which regions harbored predators. The emerging picture is clear: the human mind did not evolve in isolation. It evolved alongside trillions of microbial partners that fine-tuned the machinery of cognition. This biological partnership extended into emotional life. Microbes play a key role in regulating inflammation, and inflammation directly affects mood. Ancestral humans living with diverse microbiomes experienced lower baseline inflammation, which supported emotional stability. The comfort felt around a fire, the calm after a shared meal, the courage during the hunt, all were subtly shaped by microbial influences that balanced brain chemistry. Without diverse microbes, stress responses could become erratic, fear heightened, or focus diminished. The emotional resilience of ancient humans was not just psychological, it was microbiological. Modern research confirms this ancient truth. When microbial diversity declines, rates of anxiety, depression, and stress disorders increase. But these modern discoveries only reflect what the ancestors once knew intuitively: the state of the gut shapes the state of the mind. childhood development offers even more striking evidence. Microbes help shape neural circuits during early life. They guide the development of the vagus nerve, the primary communication line between gut and brain. They influence how the brain responds to novelty, threat, and comfort. Infants who received ancestral microbial exposures, through birth, breastfeeding, soil contact, and fermented foods, grew up with stronger emotional regulation systems. Their brains learned resilience, shaped by microbial training. But infants deprived of these exposures often carried heightened stress responses, more inflammation, and altered emotional wiring. As modern life broke the ancient contract, this microbial guidance system weakened. With fewer ancestral microbes, the brain lost some of the signals it once relied on. Neurotransmitter production patterns shifted. Emotional regulation became more fragile. Conditions like anxiety and depression rose not only because of modern stress or cultural pressures, but because the biological foundation of emotional balance, the microbial foundation, had eroded. Social behavior was also deeply intertwined with microbiomes. Studies show that microbes influence oxytocin pathways, promoting bonding, trust, and cooperation. In ancient communities, strong microbial diversity supported strong social cohesion. Humans who lived together shared microbes, and shared microbes reinforced social connection. A tribe was not only a cultural unit, it was a microbial one. Those who ate together, lived together, and touched the same soil developed synchronized microbial profiles that influenced group behavior and unity. The modern world disrupted this as well. With sterilized environments, processed foods, and fewer shared meals, people lost microbial synchrony. Loneliness, social fragmentation, and emotional isolation correlate with alterations in microbial diversity. The invisible threads that once tied minds together through shared microbial ecosystems have thinned. Microbes even played a role in shaping creativity and problem-solving. By influencing neurotransmission, microbial metabolites supported flexible thinking, curiosity, and exploration, traits that defined human evolution. The same molecules that microbes produced to stabilize their own ecosystems also nurtured the neural pathways that allowed humans to imagine, invent, and adapt. It is no coincidence that humanity's greatest strengths, imagination and cooperation, evolved alongside rich microbial diversity. But as these microbes vanished, the mind changed too. The modern brain, disconnected from the microbial signals that once shaped it, struggles in new ways. Chronic inflammation affects mood. Reduced microbial diversity impacts neurotransmitter balance. Stress responses escalate more easily. The emotional resilience of ancient humans, built through continuous microbial engagement, is harder to find in a world where microbes are often treated as threats instead of partners. This section closes with a realization that transforms how we understand ourselves. Human thoughts, emotions, fears, joys, and connections are not solely the product of neurons firing in isolation. They are the echoes of a long evolutionary conversation between brain and microbiome, a conversation that shaped what it means to be human. The microbes that once built our minds are disappearing, and with them, some of the emotional and psychological strengths our ancestors carried. The next chapter will explore whether this ancient harmony can be restored, and how modern science, traditional practices, and rewilding efforts seek to rebuild the microbial worlds that once shaped both our bodies and our minds. As the modern world began to recognize the consequences of microbial loss, a quiet movement emerged, not out of nostalgia, but out of necessity. Scientists, doctors, ancestral cultures, and everyday individuals started asking a profound question: if the ancient contract between humans and microbes has been broken, can it be repaired? And if so, how? This search for answers gave rise to a new idea, one both simple and revolutionary: to rewild the human body. To reintroduce diversity, restore lost microbial partners, and rebuild internal ecosystems weakened by generations of modern living. Rewilding did not begin in laboratories. Its earliest seeds came from the study of hunter-gatherer groups around the world whose lifestyles remained closely connected to nature. Scientists discovered that these communities carried far richer and more diverse microbiomes than industrialized populations, including microbial species that modern humans no longer had at all. Their diets, filled with wild plants, fibrous roots, fermented foods, and natural exposure to soil and animals, maintained a living snapshot of what human microbiomes once looked like. Their immune systems were strong, allergies were rare, and chronic diseases almost non-existent. These findings sparked a realization: the industrialized world had not evolved beyond microbial diversity, it had abandoned it. The first efforts to rewild the human body focused on diet. People began turning back to fermented foods: kimchi, sauerkraut, kefir, kombucha, yogurt, miso, tempeh, and traditional sourdough. These foods, once staples of ancient life, carried living bacteria capable of reintroducing diversity to the gut. Fermentation traditions revived in kitchens around the world, not just as culinary pursuits, but as acts of biological restoration. Fiber-rich diets became another cornerstone of rewilding. Modern diets lack the plant fibers that ancestral microbes depend on. Rewilding encourages the consumption of whole fruits, vegetables, legumes, nuts, seeds, and unprocessed grains, foods that feed beneficial microbes and rebuild lost ecosystems inside the gut. As people adopted these diets, scientists observed increases in microbial diversity, improvements in digestion, reduced inflammation, and strengthened immunity. But food alone could not rebuild what had been lost. Environmental exposure also played a critical role. Rewilding encouraged people to touch the earth again, to garden, walk barefoot, spend time in forests, swim in natural bodies of water, and interact with animals. These interactions reintroduced environmental microbes that had shaped human biology for millennia. Studies showed that time spent in nature reduced inflammation, improved mood, and increased beneficial microbial diversity on the skin and in the gut. Children who played outdoors regularly had healthier immune systems than those confined to sterile indoor spaces. The movement also extended to birth practices. Hospitals began exploring ways to support microbial inheritance for babies born through C-section, such as maternal microbial swabbing. Skin-to-skin contact, breastfeeding, and reduced prenatal antibiotic use became essential components of early life microbial restoration. These interventions worked to restore the microbial bridge that had broken in the modern era. Some scientists pursued even more radical approaches. Fecal microbiota transplants, transferring gut microbes from healthy donors to patients, showed remarkable promise. They cured severe infections, reduced chronic inflammation, and helped restore microbial diversity in extreme cases. Though still developing, this therapy hinted at a profound truth: microbial ecosystems could be rebuilt when given the right inputs and environment. Beyond medicine, communities revived ancestral practices unintentionally aligned with rewilding principles. Farmers' markets brought people closer to soil-grown foods. Homesteading movements embraced fermentation, gardening, and natural living. Outdoor schools encouraged children to learn in forests rather than classrooms. Even urban parks and green roofs became sanctuaries for environmental microbial exposure within cities. Yet rewilding is not simply a return to ancient life. It is a conscious blend of modern knowledge and ancestral wisdom. It acknowledges that humans cannot abandon antibiotics, sanitation, or technological progress, but they also cannot afford to abandon microbial diversity. The challenge is finding balance, embracing practices that restore microbial ecosystems while maintaining the safety and benefits of modern society. Rewilding reveals a deeper philosophical question as well: what does it mean to be human in a world disconnected from the natural microbial systems that shaped us? Modern humans often live in environments engineered for convenience and cleanliness, yet these same environments may weaken the very biology that makes us resilient. Rewilding suggests that reconnection with nature, through food, soil, water, or touch, is not just nostalgic, but biologically essential. As people adopt rewilding practices, they often notice profound changes. Improved digestion. Better sleep. Enhanced mood. Increased energy. Reduced allergies. Stronger immunity. These changes are not magic, they are the result of microbial restoration, of reintroducing the biological allies that once sustained human health. Still, rewilding is not without limitations. Some microbial species lost from industrialized populations are truly extinct within those bodies. They cannot be restored through diet or environmental exposure alone. Their ecosystems, wild soils, ancient forests, ancestral diets, no longer exist in the form they did thousands of years ago. Rewilding must accept that some parts of the ancient microbial landscape are gone forever. But this does not diminish its importance. Rewilding offers something more powerful than a perfect restoration, it offers a path forward. It recognizes that humans are ecosystems and that ecosystems must be nurtured. It challenges the sterile ideal and invites people to rethink their relationship with the microbial world. It shows that microbial diversity is not simply a relic of the past, but a foundation for future health. This section closes with a sense of possibility. The ancient symbiosis may never be fully restored, but it can be renewed. Humans can rebuild microbial diversity with intention, reconnect with nature, revive ancestral food traditions, and foster environments where beneficial microbes thrive. The next chapter will reveal that this microbial story extends far beyond human bodies, that across the planet, ecosystems everywhere are losing their microbial richness, creating consequences even more profound than those unfolding within us. Long before humans realized their own microbial worlds were collapsing, the planet had already begun experiencing a similar decline. Microbes are not only inhabitants of the human body, they are the engineers of the biosphere, the quiet chemists who sustain soil fertility, regulate oceans, recycle nutrients, and support all forms of life. As forests fell, oceans warmed, soils eroded, and ecosystems fractured, microbial communities across the earth began to weaken. The decline was subtle, invisible to most eyes, yet it created shockwaves through the natural world. The story of microbial loss was not just a human crisis, it was a planetary one. Earth's soils are among the richest microbial ecosystems ever known, containing billions of microorganisms in a single teaspoon. These microbes break down organic matter, release nutrients for plants, store carbon, and stabilize ecosystems. But industrial agriculture disrupted this balance. Heavy plowing exposed soil microbes to oxygen, causing their rapid death. Chemical fertilizers replaced microbial activity rather than supporting it. Pesticides killed not only pests, but beneficial microbial species essential for soil structure. Over time, soils became biologically impoverished, losing the microbial richness that had sustained plant life for millions of years. This microbial decline has consequences far beyond agriculture. When soil microbes disappear, plants struggle to grow without artificial inputs. Carbon that was once stored underground escapes into the atmosphere, accelerating climate change. Erosion increases, landscapes degrade, and biodiversity collapses. The Earth's microbial heart weakens, and with it weakens the foundation of terrestrial ecosystems. The oceans, too, face a microbial crisis. Marine microbes produce over half of the oxygen in the atmosphere. They cycle nutrients that feed fish, whales, and microscopic plankton. They regulate carbon absorption and shape climate patterns. But warming waters, acidification, pollution, and plastic contamination have destabilized these microbial systems. Certain species bloom uncontrollably, creating harmful algal blooms that suffocate marine life. Others vanish quietly, leaving gaps in oceanic nutrient cycles. The microbial choreography that once sustained the seas is faltering. Even the atmosphere carries microbial life, bacteria, fungal spores, and viral particles drifting across continents. These airborne microbes influence cloud formation, weather systems, and even rainfall patterns. But as land and ocean ecosystems lose microbial diversity, the composition of airborne microbes shifts as well. Entire atmospheric patterns may be subtly changing in response. Animals, too, experience microbial decline. Species raised in captivity or industrial environments lose the diverse microbiomes that wild populations maintain. Monoculture farming strips livestock of ancestral microbes, making them more vulnerable to disease and increasing reliance on antibiotics, which further disrupt microbial ecosystems. Wildlife subjected to habitat loss, pollution, and climate change also suffers internal microbial collapse. When an animal's world shrinks, so does its microbiome. Plants rely on microbial partners as deeply as humans do. Mycorrhizal fungi connect roots across forests, enabling nutrient sharing and communication between trees. Nitrogen-fixing bacteria enable legumes to grow in poor soils. Yet deforestation, pollution, and agricultural chemicals break these partnerships. Trees growing in degraded land lack the microbial allies they once depended on. Fields farmed intensively lose the underground fungal networks that once stabilized them. Microbial loneliness becomes a new form of ecological stress. These global patterns reveal a profound truth: microbial decline is not an isolated biological phenomenon, it is a symptom of environmental collapse. Every disruption humans cause in the natural world ripples downward to microscopic scales. When forests fall, their microbial layers vanish. When rivers are polluted, aquatic microbes die. When the climate shifts, microbial balances destabilize. The planetary microbiome reflects the health of Earth itself. This microbial decline also feeds back into human systems. Soil degradation demands more chemicals to grow food, which further harms soil microbes, a cycle that deepens both ecological and microbial loss. Oceanic microbial imbalances worsen climate change, which then affects agriculture, disease patterns, and global stability. Airborne microbial shifts influence allergies, respiratory diseases, and even weather. The collapse of global microbial diversity threatens both environmental and human health simultaneously. Yet even as this crisis unfolds, pockets of microbial resilience persist. Undisturbed forests still contain ancient microbial networks, green cathedrals of biodiversity. Traditional farming practices, such as crop rotation and composting, nurture soil microbes instead of erasing them. Protected coral reefs, rewilded landscapes, and regenerative agriculture projects show that microbial ecosystems can recover when given space and time. Nature remembers how to heal, but only if humans allow the conditions needed for restoration. A growing movement in ecology focuses on preserving microbial diversity the same way conservationists protect endangered animals. Scientists collect microbial samples from remote regions, storing them in biobanks that act like microbial arks. These preserved species may one day help restore soils, oceans, and even human microbiomes. Indigenous communities also hold knowledge of natural ecosystems and farming methods that support microbial health, knowledge increasingly valued by modern environmental science. Still, the global picture remains sobering. The Earth's microbial systems have been shaped over billions of years, yet human activity has altered them in just a few centuries. The loss of microbial diversity weakens the planet's resilience, making ecosystems more vulnerable to collapse. Just as human bodies suffer when internal ecosystems shrink, the Earth suffers when its microbial foundations erode. This section closes with a realization that deepens the emotional and philosophical weight of the story. Humans are not only losing microbes within themselves, they are losing microbes across the entire planet. The microbial decline in human bodies is a reflection of a broader environmental crisis. The ancient symbiosis that supported life on Earth is faltering, not because microbes have changed, but because the conditions that once nurtured them have vanished. The next chapter will explore what this means for the future, which microbes may survive the modern world, which may disappear forever, and what the microbial landscape of tomorrow might look like in a world shaped by human hands. As the modern world accelerates into the future, one question looms over both science and philosophy: which microbes will endure the next century of human activity, and which will vanish forever? The story of humanity's microbial allies is no longer just about loss, it is about survival. In a world shaped by antibiotics, sanitized environments, processed diets, climate change, and globalized ecosystems, only certain microbial species will persist. These survivors will define the microbial landscape of future generations, shaping health, behavior, and even evolution in ways we cannot yet fully imagine. To understand the fate of microbes, we must first recognize the forces shaping their future. Modernity exerts immense pressure on the microbial world. Antibiotics eliminate entire bacterial kingdoms within hours. Industrial foods starve fiber-dependent species. Chlorinated water blocks environmental microbes from entering the body. Urban air reduces microbial diversity. Climate change alters the habitats of soil and ocean microbes alike. These pressures select for certain traits: hardiness, adaptability, and resistance, while filtering out species that rely on delicate ecological balances. Some microbes will not survive these changes. Many ancestral bacteria that once thrived in rugged, fiber-rich guts have already disappeared from industrialized populations. Others linger only in remote communities, gradually vanishing as traditional lifestyles fade. Species dependent on diverse plant diets or natural environmental exposure face intense extinction risk as global diets homogenize and people spend more time indoors. Without major environmental or cultural shifts, these microbes may vanish entirely, surviving only in scientific archives. Yet not all microbes weaken in the face of modernity. Some flourish. The first group of survivors includes antibiotic-resistant bacteria. Evolution drives these microbes to adapt quickly, exchanging genetic material that grants immunity against even the strongest drugs. These species, some harmless, others dangerous, will undoubtedly persist, growing stronger as antibiotic pressure intensifies. Their resilience may become both a challenge and a reminder of microbial tenacity. Another group poised to survive are microbes that thrive in low-fiber, high-sugar environments, species adapted to modern diets. These bacteria multiply rapidly when fed processed foods, outcompeting ancestral microbes. Though not always beneficial, their ability to adapt ensures their survival in industrialized guts. They have evolved to endure inflammation, metabolic imbalance, and chemical additives, becoming the new dominant species in many human bodies. Environmental microbes will also shift. Soil bacteria sensitive to disruption may disappear, but hardy species capable of surviving pollution, drought, and degraded ecosystems will remain. These microbes will redefine soil health in a world where traditional nutrient cycles have been altered. Similarly, in oceans, resilient microbes will adapt to acidic waters and fluctuating temperatures, forming new ecological networks while more fragile species die off. The microbial world of the future will not simply be smaller, it will be different. But perhaps the most surprising survivors will be the microbes preserved by intentional human effort. As awareness of microbial decline grows, scientists are racing to collect and store microbial species from around the world. These archives, known as biobanks, act as microbial time capsules, preserving strains from hunter-gatherer communities, remote ecosystems, ancient fermented foods, and traditional soils. These microbes may one day help restore lost diversity through medical treatments, environmental rehabilitation, or even future rewilding projects. Human culture will also play a role in shaping microbial futures. Fermentation, once fading, is returning as people rediscover traditional foods rich in living microbes. Homemade sourdough, kombucha, kimchi, and kefir carry resilient species that thrive in modern kitchens. These microbes, preserved through cultural memory rather than nature, may become some of the last remnants of ancestral microbial lineages. If society continues to embrace fermented foods, these microbial allies may survive far longer than their wild counterparts. The future of microbes is also tied to childbirth trends. As medical communities explore microbial restoration techniques for infants born via C-section, some ancestral species may be passed down intentionally through maternal microbial swabbing, breastfeeding, and low-intervention birth practices. These efforts may help preserve species on the brink of disappearance, keeping lines of microbial inheritance alive even as the modern world continues to evolve. New technologies will further influence microbial survival. Advances in genetic engineering, synthetic biology, and microbiome therapeutics could allow humans to design microbes tailored for specific environmental or medical purposes. Engineered microbes might one day replace species lost to extinction, performing roles essential for human health. These solutions blur the line between natural evolution and human design, opening a future where microbial survival depends not on nature alone, but on scientific intervention. Still, some microbes will endure for reasons beyond technology or culture, because life itself is resilient. Microbes have survived mass extinctions, ice ages, volcanic eruptions, and cosmic events. Their ability to mutate, adapt, and evolve far outpaces that of any other organism on Earth. Even as modernity reshapes ecosystems, microbial life will continue to find pathways to persistence. Some species may retreat into hidden niches, the deep ocean, untouched forests, underground soil layers, waiting for opportunities to recolonize when conditions improve. But what does it mean for humanity if only the hardiest microbes remain? The consequences will be profound. A future dominated by resilient but less diverse microbial ecosystems may lead to increased chronic illness, weakened immunity, and altered emotional landscapes. Microbiomes that lack ancestral complexity may struggle to support balanced inflammation, efficient metabolism, and stable mental health. The internal world of future humans may be more reactive, more fragile, more dependent on medical intervention. At the same time, the rise of engineered microbes offers hope. Just as humans once unknowingly shaped microbial evolution through fire, fermentation, and agriculture, they may consciously shape microbial futures through biotechnological innovation. The microbes that survive modernity may therefore fall into two categories: natural survivors shaped by evolution, and engineered organisms shaped by human intention. This blending of biology and design raises philosophical questions. Are humans still part of nature if their internal ecosystems are curated by technology? Can a microbiome shaped by probiotics, engineered species, and restored ancestral strains replicate the richness of ancient microbial worlds? Or will the future human body become a hybrid ecosystem, part natural, part constructed? Regardless of how these questions unfold, one truth remains clear: the microbial world will not disappear. It will change, adapt, and re-emerge in forms shaped by both survival pressure and human influence. What disappears may not be microbes themselves, but the specific species that once walked alongside humanity through 100,000 years of shared evolution. This section closes with a sense of both caution and possibility. Some microbes will endure the fires of modernity. Others will fade into history. And humanity, no longer passive recipients of microbial inheritance, will play a defining role in determining which species survive. The next and final chapter will explore what this means for the ancient pact, and whether humans, standing at a crossroads, can choose to rebuild a new symbiosis for the future. For nearly 100,000 years, the relationship between humans and microbes shaped the story of life more profoundly than any invention, empire, or revolution. It was a partnership that evolved in silence, woven into breath, skin, soil, and food. It guided human immunity, emotions, metabolism, and even behavior. It helped early humans survive unforgiving landscapes, adapt to new environments, and thrive as a species. But in the last blink of evolutionary time, that relationship changed. It frayed, weakened, and in many ways, broke. Now, humanity stands at an unprecedented moment, a crossroads where the future of this ancient partnership depends not on evolution or chance, but on deliberate choice. This epilogue explores that choice, whether humans will continue drifting away from microbial diversity, or consciously rebuild a new symbiosis shaped by understanding rather than instinct. To understand the weight of this choice, imagine the span of human evolution compressed into a single day. For almost the entire day, from dawn until minutes before midnight, humans lived in harmony with the microbial world. They ate wild plants, gathered roots, hunted animals, fermented food, gave birth naturally, slept close to the earth, shared microbial signatures with their communities, and existed within ecosystems that supported microbial richness. Only in the last few seconds before midnight did everything change. Modern life, with all its brilliance and innovation, rewrote the terms of the ancient contract. And yet, it did not destroy the possibility of a new one. The choice begins with awareness. For so long, microbes were invisible, either unknown or misunderstood. Modern science, however, offers a clearer vision. It reveals that microbes are not merely passengers in the human body, they are foundational partners. They help regulate emotions, teach the immune system, break down food, produce vital compounds, and influence the body in ways people are only beginning to grasp. The recognition of this truth represents the first step toward a new relationship, one grounded not in fear or ignorance, but in knowledge and respect. The next step is intention. Humans did not intentionally break the ancient pact. They did not mean to starve fiber-loving microbes with processed diets, nor did they seek to sterilize their internal worlds through excessive sanitation. These changes came from the desire for safety, longevity, and convenience. But now that the consequences are known, intention becomes crucial. People can choose which aspects of modern life to preserve and which to rethink. They can choose to protect microbial diversity rather than inadvertently harm it. This intentional shift begins in everyday actions. A person who chooses fermented foods over sterile snacks reintroduces living microbes into their gut. Someone who walks barefoot on soil or tends a garden rebuilds the microbial connection to the earth. A mother who breastfeeds or seeks microbial restoration after a C-section passes vital microbes to her child. A family that embraces fiber-rich meals supports species that have survived since ancient times. Small decisions, grounded in understanding, can begin to heal the invisible ecosystems within. But the choice cannot be individual alone, it must also be societal. Modern medicine, for example, stands at a delicate intersection. It must preserve the power of life-saving antibiotics while using them with greater wisdom. It must continue protecting patients through sanitation while recognizing that not every microbe is a threat. New guidelines, emerging research, and patient education become tools for balancing safety with biological harmony. Agriculture, too, faces a crossroads. Regenerative farming, crop rotation, reduced chemical use, and soil restoration can rebuild microbial life beneath the ground. These practices not only benefit the environment but indirectly influence the microbes that eventually enter human bodies. The soil microbiome, once thriving and diverse, can become vibrant again, but only if its importance is recognized and protected. Urban planning offers another realm of possibility. Cities filled with green spaces, community gardens, natural parks, and clean rivers allow urban populations to reconnect with environmental microbes. Schools designed with outdoor learning spaces help children develop stronger microbiomes from early life. These design choices are not merely aesthetic, they are biological investments in future generations. Technology, once seen as a force separating humans from nature, may become a partner in microbial restoration. Advances in microbiome science could eventually allow scientists to revive extinct species, design probiotics tailored to individual needs, and rebuild internal ecosystems with precision. The future may hold microbial therapies that restore what was once lost, complementing diet, lifestyle, and environmental exposure. And yet, despite scientific progress, one truth remains: humans must rediscover humility. Microbes were here billions of years before the first humans walked the earth. They shaped the atmosphere, prepared the soil, and made complex life possible. They are not simply tools to be managed or problems to be solved. They are ancient partners in the story of life. The arrogance of modernity, the belief that humans could dominate nature without consequence, lies at the heart of microbial decline. The future requires a new mindset, one where humans recognize themselves not as separate from microbial ecosystems, but as part of them. This leads to the deepest aspect of the choice: emotional and philosophical reconnection. Modern societies often operate under the illusion of control. Clean lines, sealed buildings, sterile surfaces, packaged foods, these create a sense of predictability, a buffer against the chaos of nature. But beneath that illusion, biological richness fades. To rebuild microbial harmony, humans must become comfortable with the idea that not everything can or should be controlled. That life thrives in diversity, in organic complexity, in ecosystems that cannot be micromanaged. Rewilding the human body, and the planet, is ultimately an act of trust in nature's wisdom, in evolution's design, in the biological intelligence that governs microbial communities far more intricate than anything humans can engineer. As this story reaches its end, imagine a newborn child of the future. Born in a world more aware of microbial necessity, this child receives a rich microbial inheritance from its mother. It grows up exploring natural spaces, eating diverse foods, touching soil, knowing both the brilliance of modern medicine and the wisdom of ancestral practices. Its immune system develops with guidance. Its microbiome flourishes. Its body becomes a living symphony of ancient and modern microbes, functioning in harmony. This vision is not utopian, it is possible. The future of human microbial symbiosis depends on the choices made today. Will societies continue down a path that narrows diversity and weakens resilience? Or will they embrace a future where microbial life is recognized as essential, protected, and nurtured? The answer lies not only in science, but in values, how humanity defines health, progress, and its place in the natural world. This chapter, this epilogue, ends with a quiet truth: the ancient contract may be broken, but the story is not over. Microbes have shaped humanity from the beginning. And now, perhaps for the first time, humanity has the chance to shape the future of that relationship with intention, humility, and hope. The last microbes are not merely survivors, they are reminders of what was lost and guides toward what can still be rebuilt. The next 100,000 years will not be written by evolution alone, but by the choices humanity makes today, choices that determine not only the fate of microbes, but the future of human life itself.